I'm a medicinal chemist by training. I've worked on a number of different fields of research looking for new small molecule therapeutics, anti-cancer, antibacterial agents, ion channels, diabetes, metabolic disorders, lots of different protein-protein interactions. But the kind of thing that ties all these together is they've all used ligand-based approaches and computational chemistry to identify small molecules to modulate proteins of interest. So I wanted to just briefly, I've got some props. So I'm going to talk about a lot about ligand docking, but I guess from the audience that maybe some people don't know too much about that. So for the purpose of today, I've got a little bit of rubbish one. I've got better ones than these now. 3D printed protein here. So ligand docking is looking at, if we have a ligand like this, or something like a drug, working out using computers how it can bind to our protein of interest and which is the best way, where's it most likely to bind and how likely is it to bind. So when I talk about ligand docking, that's what I mean, these little things to the big proteins. So when I started my PhD here in Leeds in 2005, we were able to do single docking, so just one of these to one protein on a local Linux machine. And we were able to use library sizes of around 50,000 ligands in this time. The inception of the ARC1 screening computer platform in 2009 and an in-house electronic high throughput screening platform called eHits meant that we were able to kind of improve on this. And when we got to ARC2 and ARC3, bringing other software from companies like Glide from Schrödinger, we were able to do up to about 100,000 dockings in a weekend. We've been able to bring in more different algorithms to work on ligand binding because all these different algorithms have their own foibles. They all have various things that they do well, various things that they don't. So we wanted to use as many as possible to get the best idea of how our ligands would bind into our protein. By the time we got to ARC3 and ARC4 at the end of the last decade, we were able to do around 1 million dockings in a weekend, and our library sizes are currently about 20 million. So we've come quite a long way from 50,000, and hopefully we can look at ways to improve on this even further in the next few years. So just to give an idea of the approach we're using, so we use a number of different computational methods, which I'm mainly going to talk about the virtual screening today. We can also look at ligand similarity or de novo design from scratch on a protein ligand complex. We can then go in the lab and synthesize our little molecules. We can then test them, and we can do iterative rounds of this to understand how our molecules are binding to our target of interest and whether they're doing what we want. So I'm going to do a little case study now for a couple of minutes, look at these insulin receptor hybrid modulators. And this is a case study of a ligand that's been developed using these methods on ARC3 and 4 in the last sort of 5 to 7 years here at Leeds. So briefly, type 2 diabetes is when people's blood sugar levels become elevated. It affects around 4 million people in the UK. It can cause serious long-term health problems like loss of vision and kidney failure. As well as this, people with diabetes are about 5 times more likely to have cardiovascular disease, and they also have a worse prognosis when diagnosed with these conditions. It costs the NHS at least £14 billion per year to treat diabetes and its complications. So we thought these insulin receptor hybrids have been shown to reduce insulin sensitivity in people who've got type 2 diabetes. It prevents their vascular system from being able to repair itself. So if we could reduce these hybrids in our blood, we could help to look at whether that could treat type 2 diabetes. So this was the inception of this case study. So we created a model of what we think our insulin receptor hybrids look like using a number of different homology modeling platforms. So obviously nowadays, we use AlphaFold. This wasn't available in 2015, so we used a number of different homology modeling servers to get a consensus of what we think our hybrids look like. And then we used hotspot mapping software to understand which regions were important for these proteins coming together to form these hybrids. So this is the screening workflow that we used on the ARC system. So we screened made a small set of 100,000 ligands of the zinc drug-like diverse set, which was produced using Omega, something that makes 3D representations of our little ligands on ARC3. At the time, this was doing about 2015, so we were really at sort of 100,000 stage. Now we can do many more than that. So this was screened in a high throughput mode using our Glide software from Schrödinger, and then the top 20,000 best looking molecules were taken forward to screen at a high precision. We did some consensus scoring with a different scoring algorithm, because we know, as I said before, that these algorithms all have their own goods and bad points. So we want to make sure that we're really trying to get a molecule that binds to our protein of interest. We want to weed out compounds that interfere with lots of different assays that have known pharmacophores that do this. So we do this at this stage before visually inspecting how our little ligands bind to our protein of interest, synthetic steric clashes, hydrogen bonds, etc. And this is when we buy the compounds. So conceptually, we have our hybrid protein structure, and as I said, we've got our idea of which bits are important for making these hybrids. If we zoom in on this little red patch, we use the computational software to identify the ligands that bind within this red patch, and we can see here that they're predicted to make interactions with one of the monomers and make a clash with the other monomer, which we think can prevent these hybrid receptors from forming. So we purchased 50 molecules in this subset, and we were able to identify this compound 4 here, which I've marked with this big purple arrow, as reducing the amount of these hybrids that we have in human cells. We were able to take this forward and use cell lines from people with diabetes to show it has exactly the same effect. So this is the graph on the left here, and that it doesn't affect expression of the proteins. This is exactly what we wanted to see. So we're really pleased with this result. We were able to do a lot more of this analysis to understand the kind of downstream effect of reducing these hybrids, and I've tried to show this in a cartoon here. So if we expect these hybrids are these little green and yellow monomers here, with homodimeric protein shown in green and yellow, this molecule looks to bind to our hybrids, preventing them from forming. And this affects the amount of this insulin receptor substrate, it reduces the amount of this kinase, increases phosphorylation of these proteins, which leads to more nitric oxide being produced in your cells. This nitric oxide is really important, has lots of cardiovascular benefits to improving vascular health. So we think that our molecule identified using these approaches is having a real positive effect on human cell health, and we hope to take it forward into mouse models. So where next? What barriers have we got at the moment? So there are groups out there who are using cloud computing to screen billions of molecules now. And there are libraries available from companies like Enamine who have got 4.5 billion small molecules within them. So obviously we're quite a long way away from that at this 20 million level, but how can we get closer to them? So our main barriers at the moment, and I think it's like you said earlier, the barriers seem to move all the time. We have problems with transferring large files between our local machines and the ARC computing cluster. We have problems with job times. How many dockings can we get done in the 48-hour windows allowed in ARC? There's a lot of variability in this, which makes it hard to understand how many to put in each library. I think all of you can say the people at ARC are really good, but there's just not enough of them to get all the help that people need going forward. And we also struggle currently with getting IT support for our local Linux machines. But that said, I think we're really making progress from where we were a decade ago to where we are now, where we can screen millions of molecules in a weekend. And hopefully within the next 5 years, we'll be upping that to hundreds of millions and we'll be able to compete with the big boys. So I'd just like to acknowledge some of the people who've done this work over the last 17 years now, seems like a long time. No, most of them aren't on this, but anyway. So these people on the left are the people who've done the case study that I just spoke about with the hybrid inhibitors. These are my supervisors when I was a postdoc and PhD student. All the people who've worked in the ARC help centre over the years. I think David was the first person I ever emailed in like 2009 about this, when I was even worse at using these machines than I am now. And I'd like to thank David Waller and Vilma Spalko for their past help with our local Linux machines and getting off the ground with this initial screening. I'd like to finally thank funders, the European Union, Diabetes UK and the British Heart Foundation, who funded me over the years, and everyone here for your attention this afternoon. I was worried I was going to run over.
I have a question for you at the end.
Okay.
I need to. So you stopped sharing, Katie?
I just closed my session. No, I didn't stop anything else.
Has it stopped? Okay,
Presentation.
so maybe I need to. Alistair. Yep. Let's close this one. Go to Zoom, just start over sharing. Where is it? This one is yours. Yeah. Thank you.
Right, thanks very much. Great, thanks very much. Yeah, it's great to be able to share in the context of people doing different research computing. I'm going to present software I've developed with others, and others have certainly helped a lot with this. Purple is my acronym in microscopy. It's all about the acronyms. And I will explain what that means, I think, at some point. So is that a pointer? That's a pointer. How do I make the clicker work? Or do I just do this? Yeah, great. Okay. So these are people and organizations, charities that have kept me in work for the last few years doing this. So I'm very grateful to them, especially working with Michelle in molecular and cell biology. So this is going up a level from what's happening at the level of a single protein and something docking to it. So when proteins are doing their jobs in cells, they're organized into different structures. How can we quantify? We can get pictures with microscopy of what those structures are, but how can we quantify what the organization is like? Often we get quite blurred pictures just because of the nature of light and microscopy, as Yehuda explained earlier with electron microscopy. And now we can go from these blurred pictures to localize individual molecules. So this is, you get individual proteins to light up, and you detect where they're lighting up, and you can reconstruct that into an image afterwards. So you go from something which looks like a round blob to something that you realize actually there's a ring structure to it, which is really interesting. And that's something that is present on the nuclei of all of the cells in your body. It's called the nuclear pore complex. And this is from data from a colleague in Germany, Jonas Ries at Empel. So we can get nice structures in cells. So one question is, how can we quantify different structures there are in cells? Another question is, how can we then make that useful for people being treated for the right disease or understanding what's going on in diseases as well? So now looking at projects with people in medicine. So it's been really great, funded by Wellcome, to get more in touch with the computing people, more in touch with the medicine people. This is in Phil Quirk's group, who's very interested in this new technology for diagnosis using this type of microscopy. This is a colorectal cancer patient tissue where we've labeled the individual proteins, and you can see there in these clusters with patterns, particularly around the edges of the cells, the membranes. And this is from kidney disease, where electron microscopy is often used for diagnosis, which is a very, in that context, in the hospital context, it can do wonders. In the hospital context, it's very expensive and very slow. So can you get down to the kind of length scales that you look at with electron microscopy much faster and cheaper? Possibly, that's what I'm trying to find out. And this is working with Oxford Nano Instruments, who produce this type of microscope as well. So it's a relatively new type of technology, certainly in terms of clinicians are concerned, because obviously things have to be validated to get anywhere near being used in that context. Because it's a relatively new type of microscopy, there isn't actually a great diversity of ways of analyzing the data yet. So this is why I'm part of developing those analyses. So how am I going to start to quantify arrangements in the cells? So one idea is, oh, sorry, accidentally hit the clicker there. So look at, so behind these light and dark patches, there are actually individual coordinates of where these proteins have lit up, and then I've reconstructed them into something like a histogram afterwards. So you get a point coordinate every time a molecule lights up. You can ask me some other time how they get them to light up, how we get them to light up. So it's not an image where there's lots of image analysis techniques. It's actually a list of coordinates, point cloud, if you like, and how we're going to analyze the point cloud for features of biology and disease. So we could look at what are the relative positions between the localizations that pop up. And this will have the advantage of, we could try single particle averaging like in electron microscopy, and people have tried to do that quite a bit. But one of the issues in this type of microscopy is you try to get everything to light up, but actually you don't get everything to light up. You get a lot of points missing out of your point cloud, which makes the, you can imagine if you try and get all these different images of single particles then to match up together, you've got a lot of information missing. So looking at something like this, looking at relative positions between them, you will still catch, when there's a certain set of relative positions that are very common, you will still catch those characteristic situations that the molecules find themselves in. So if we just plot the XY separations between pairs of these molecules, we don't search out too far because the number of pairs of molecules explodes. But you start to see these characteristic peaks in the distance distribution between molecules, which is really nice. So this very sharp peak here is where the same molecule will actually repeatedly blink. That's the very sharp one. And then you've got characteristic distances like here and here between the molecules. And you've got a constant kind of increasing background, which is what you expect from a 2D distribution as you go further out in distance. So what can you do with that distribution then? Well, you can think, well, what have I really got? It looks a bit like a donut. Maybe there's defined points around a ring. Maybe it has some kind of symmetry. So you can develop a model for what the characteristic distances would look like if it had certain symmetries. So you can model symmetric rings, again, with these different peaks for different characteristics that I mentioned. So B, C, D, I can't see from here very well, and E, they're these characteristic distances across the octagon in this case. And you can make a parametric model for what you would expect those components of the distance distribution to look like, and you can fit it. And you can fit it for all of your different models. So 5 to 11-fold symmetries are in there. And then you need some way of distinguishing which model actually gave you the best result. And this is with the a KK information criterion that I used here, which is quite nice because it tells you how likely a model is to be the best model whilst penalizing you for overfitting. So if you just put loads and loads of parameters in there so it definitely fits, this information criterion will say you cheated there, you can't have that. And lower number wins. So fortunately, this 8-fold model came out as the best model, and the nuclear pore complex is 8-fold. So that was good. And you can transform these information criterion numbers into relative likelihoods as well over there. And because you're fitting these distance distributions, they all come from the diameter of the ring as well. So you can pop out things like the diameter that you fitted it to, and this comes out exactly right for this complex as well. So this is a way of spotting the characteristic distances between individual proteins, so not within a protein, like Katie was looking at, but between different proteins, and what are the distances there, and what do they mean? So now into disease. So this is part of a kidney. This is part of a glomerulus, and there are these capillaries that go here. And what electron microscopy is used for a lot in kidney disease is trying to distinguish between diseases. And it's used because there are differences that you see at a very small scale with electron microscopy that you can't currently see with anything else. So we think we might get close with being able to localize single molecules with fluorescence and seeing what the distribution is there. Are there any characteristic distances? That kind of thing. So this is Ollie, who's actually in the CDT looking at cancer primarily, but along the way, he's developed this little bit of software that allows me to manually segment segments of these capillary membranes where we're most likely to see the pattern, if there is one. And we're just starting to see these distances occur. So what happens here is quite interesting. So the tiny blobs here, these tiny light blue blobs here, they're called foot processes of these big blue cells. And they're a bit like fingers that they wrap around these capillaries, but they can be pointing in any different direction. So actually we won't see a single peak here because that would just be if these feet were pointing exactly that way, say, but actually as they move across in angle, we'll see peaks move around. So this is just preliminary data, but we're seeing these peaks here, and there are diseases where these feet are basically obliterated. So then we expect to be able to tell the difference between healthy and diseased in that case. That's the kind of thing starting to look at with this software, looking at relative positions and distances. Oh, I haven't told you what the acronym is. So purple is pattern extraction from relative positions of localizations. And is that 1 minute or is it finished?
1 minute.
Okay, so along the way, I've had lots of help. Joanna in computing has been particularly helpful in helping me to start to make it usable, because of course we published it and said other people can use this, and it's uses terminal input at the moment, which won't be very friendly for a lot of people, and takes you through model fitting in Jupyter notebooks. But we would like it to be easier for people to use than that and a bit more interactive. And there's lots of new analyses to do on this point cloud type data, which, as I said, is really undeveloped in this field, but is really needed so that when super resolution microscopy was invented and it won a Nobel Prize as well, everyone thought this is going to be amazing. We're going to discover so much, but actually not that much has materialized. And it's partly because there's this bottleneck at the analysis stage. So research computing is very important. And so Alex has been helping point me in some really good directions in this as well. It's been great to meet a couple of people here today as well. Thank you very much.
Next.
Well, if you've got any questions, please email me.
I'll have one. I'll have one later. I'll seek you. Now it's Michael Zulsinski. Is that correct? Did I say right? Nice. It's a PDF. Oh, how do we do? Is this one? Enter PDF full screen. Yeah. Now I need to go back to Zoom and the procedure here. I'll stop sharing. Yes.
Yeah, it's working well. Okay, so hi everyone. So I'm Michael, and I'm going to present a bit different work than the two previous speakers. And what I'm going to talk about is transcriptomic analysis of temporal artery biopsies to identify novel pathogenic pathways in giant cell arteritis. So just to give a little bit of background, giant cell arteritis is the most common form of vasculitis in people over 50 years old. And it's characterized by inflammation in medium and large vessels. And the pathological changes that occur in GCA patients include the inflammatory infiltrates through the arterial wall. Like as you can see on the picture here, there's three layers of the arterial wall, intima, media, and adventitia. And what we're interested the most, what I'm interested the most in my project is to look at different phenotypes, which the phenotypes mean different inflammatory infiltrates. So basically, we look at the distribution of the inflammation through the arterial wall. And there's a scoring system that was developed by a team in Spain, which was described in this paper. And here you can see some examples of the different patterns. Those are basically histological images of the arterial wall with showing different inflammatory patterns. And the main aim of my project is to use transcriptomic data obtained from giant cell arteritis positive patients, along with some histological images and also clinical characteristics to, first of all, gain a better understanding of the pathogenesis of GCA and ideally, and hopefully in the end, to identify some new candidates, either transcripts or pathways for therapeutic targeting. And here's the list of three objectives that I'm going to sort of showcase a bit of my PhD project. And here are the three objectives that I'm going to show the results for. So they are to assess confounding influence of some of the clinical variables, such as age, gender, and exposure to steroids. And also the main sort of analysis is to examine associations of transcripts level with histological phenotypes, which are basically the different inflammatory patterns. And also then at the end to identify a list of transcripts that relates to these different GCA subtypes and outcomes. And here's the summary of the data that I use. So as I said, the main type of data that I use is the gene expression data. And we have a nice cohort of about 80 unique samples that are derived from GCA positive patients. Oh, sorry. And yeah, and what's important to highlight here is that all the patients are GCA positive, so we don't have any healthy controls for now. So because the main focus of this study is to basically the main focus is on the pathogenesis of GCA and on looking at different subtypes. But hopefully later this year we'll get some healthy samples as well. Yes, so all the patients the data was sequenced in an RNA-seq experiment, and then it was processed using a pipeline that I developed on the HPC. And what you can see on the bottom part of the slide, this is the processing of the histological data that we get. So as I said earlier on, is that we're interested in different histological subtypes. So all the patients had temporal artery biopsy, and then this tissue went through some serial sectioning. So it was basically cut in pieces, and then it was stained and scored by a histopathologist, like according to this scoring system that I mentioned before. And also what is not shown on this slide is we also have a lot of clinical information about the patients. And just for the sake of reproducibility, here are the methods that I use to generate the results that I'm going to present. So basically, just to go quickly through them. Yes, I used Mann-Whitney tests to assess the influence of the confounding influence of clinical variables. Then I used principal components analysis for dimensionality reduction and visualization. And then for the main part of analysis, which means for looking at the association of different GCA subtypes with transcript levels, I also use either Mann-Whitney or the DEC2 package in R. And then lastly, but very important, I applied multiple testing correction using false discovery rate. So here are the results. So for the first part, which was to assess the influence of steroids treatment on gene expression. So we know from other studies and that clinically we would expect that the steroids exposure will influence the gene expression, because it has been shown in many other studies. So we wanted to make sure that this is not the case in our study as well. So what you can see in this frequency diagram is that the number of days that patients stayed on steroids and the frequency. And as you can see, all patients stayed on a relatively short period of time on steroids, up to like about 2 weeks. And then to see whether there's a difference in the expression profiles of those that stayed on a short time period versus those on a longer period, I basically did like a statistical testing comparison. And the way I defined the shorter exposure and longer exposure is basically the shorter is either the first half of the distribution or the first quartile. And then respectively, the longer exposure is the second half or the first quartile. And the reason to use the quartiles was just to have more extreme values for the statistical testing and for the comparison. And in this table, you can see the results. And after applying multiple testing correction, which is in the last column, we do not have any transcripts that were statistically significant, which we can conclude that we don't have any evidence to say that steroid exposure influenced the expression of gene expression in our cohort. And that's probably due to the fact that it's a relatively short period of time that the patients stay in steroids. So moving on, so another also that can be seen as a compounding influence, that I looked at was the influence of gender. And these results were quite surprising. because so when I did my first PCA plot, like when I did principal components analysis, and so the first plot with the whole, when I put the whole data as input, which corresponds to this plot on the left, and I was quite surprised to see that there's a clear separation between the male and female samples. So that made me think that there may be something that drives this separation, and that maybe that probably it's somewhere on the X and Y chromosomes. So to test that, to check if this is the case, I removed the sex chromosomes, X and Y, and I only put the autosomal chromosomes as an input. And this is what is shown in the middle plot. And as you can see, the separation disappeared. So that suggests that the separation is driven by some of the genes that are on either X or Y chromosomes. And then the third plot is just it's just X versus Y. It's just X and Y, so then you would expect them to be clustered separately. And they are. And now, finally, moving to the main output of my analysis. So this is the table with the associations with clinical and histological phenotypes. So this is basically results of statistical testing. Same as previously in the last column, these are the results of the after multiple testing correction. And as you can see, for clinical variables, we do not have much statistically significant results, except for a little bit for gender. While for some of the histological phenotypes, we did find a lot of statistically significant transcripts, which find strong associations. And then to explore this further, for some of these phenotypes, this is still an ongoing work where I'm trying to sort of narrow down this long list of transcripts that were found to be statistically significant. And here I just look at some overlaps between, so from clinical perspective, what are the phenotypes that make sense to look overlaps between. And here are just some results from this, but this is still an ongoing work. So just to wrap up everything, so as I showed, we did not find any statistical evidence to say that there's influence of steroids treatment or age on gene expression, but we did find some evidence to say that gender is very likely to be a confounding factor. And then there were 3 of the histological phenotypes that have the strongest associations. And on the other hand, we did not find any associations with the clinical variables. And in terms of the prospective work, I'm going to further interpret the results that I got. And also we do have a single cell data set that I can also make use of. And that's pretty much the end. So I'd just like to thank my supervisors and other members of the team that helped me with this research. And thank you for your attention.
I'm suspected to say, that's the 3rd time I hear about your project. It's always fascinating. And we love transcriptomics, right? So Dhananjay, please. Did I say your name correctly? Sorry if not. Did I pronounce correctly your name? Yeah. Dhananjay. I need to stop sharing this. Oh no, not the same again. This mouse is terrible. This one, right? Thank you so much.
Hello, my name is Dhananjay. Today I'm going to talk about screening of small molecular emitter for LOX1 scavenger receptors. In this study, I'm not going to go more detail in biology and these computational things because I'm not a computational person. I'm mostly working on biological things, so I don't know programming or anything. So I'll be just go through overview of how I screen small molecules using different approaches. So for my work, we use LOX1 scavenger receptors. So this LOX1 scavenger receptor is pro-atherogenic molecule. This LOX1 is responsible for heart disease, diabetes, stroke, and metabolic syndromes. This LOX1 scavenger receptor is type 2 membrane glycoprotein, and this LOX1 is a homodimer in structure, which consists of C-type platinum-like fold, which contains 131 amino acid, which is present on here on the top of this, and number 1. And second one is then extracellular stalk region, which contains 82 amino acids, which is this region. And third one is present inside the membrane, which is called as a single transmembrane region, which contains 26 amino acids. And then last one is the N-terminal cytoplasmic region, which contains 34 amino acids, which is present inside the cytoplasmic region. In this work, we mostly focus on this C-type platinum-like fold, which contain 131 amino acids. This is our main target. So then we take that, we download the protein structure from PDB database, which contain 5 disulfide bonds. And this 4 disulfide bond is responsible for making stable structure. And if you see this, the 5th one disulfide bond, cysteine 140, is responsible for formation of dimer in structure. This dimer structure is responsible for binding of oxidized LDL. So if you see this one is oxidized LDL, and if the 3 molecules of CTLD doubles come together and interact with the oxidized LDL, which is from the responsible for atherosclerosis. So we mostly focus on the CTLD domain to inhibit the interaction between this oxidized LDL and CTLD. So for this work, we use different screening approach for such as physiochemical property filtering, 2D screening, pharmacophore modeling, molecular docking, and PENS remover, clustering, then MD simulation, as well as free energy calculation. Firstly, we download the protein structure which from PDB, which is have 6019 PDB structure, which contains one inhibitor, which is a BI0115, and this protein contains A chain and B chain with 2 alpha helix, which is showing a red color, this red color, and 5 beta sheets are there, which show in a yellow color. For this screening, we use NPAS and Maybridge compound library, which contains more than 84,000 compound, and we screen this 84,000 compound using physiochemical property, and then after we screen this compound using 2D similarity screening using Temexon software. Then after we generate the ligand-based pharmacophore modeling. For ligand-based pharmacophore modeling, we use the reference compound, which is I shown in previous slide, this one reference compound bio0115 as a reference compound. Using that compound, we generate the pharmacophore model. In that, this selected compound contain a reference compound contain hydrophobic features, which is showing yellow color, this one, and hydrogen bond features showing green color. If you see, this one is the hydrogen donor feature, and hydrogen acceptor feature, which is showing this red color, which is this hydrogen acceptor, and the excluded volume, this is excluded volume. So based on this reference compound, we generate the pharmacophore, and we select the three pharmacophore model, and all these for 3 pharmacophore model contains hydrophobic feature, hydrogen bond donor feature, hydrogen acceptor feature, as well as which contain hydrogen also, and aromatic ring. These are aromatic rings. Based on these 3 pharmacophore model, we screen down the compound and we select the compound which is having pharmacophore fit score is more than 50. So if you see in this green box, we select this compound for further study. Then after we go for molecular docking and clustering. For molecular docking, we use Autodock software. After the using Autodock structure, we select the compound which is having binding energy more than -7. So there is 49 compound we find which binding energy is more than -7. We select this 49 compound from clustering. For clustering, we find the 17th cluster. From this 17th cluster, we select the 2 cluster, and each cluster contains 4 compound. If you see this CTLD domain, these are the residues involved in the interaction with oxidized LDL on the surface of that protein. And if you see that this is a basic spine, this basic spine also responsible for interaction with oxidized LDL. So then after we check the interaction between protein, after docking, we check the interaction between protein and ligand. Some amino acid involved in interaction with A chain residues. Some compound involved with the interaction with B chain residue, and some compound will be responsible interaction with A chain and B chain. If you see in this image, this compound is involved in the interaction with A chain amino acids. And if you see this compound, which name is here, NPC 4759, which is responsible for interact with chain B amino acids. And if you see this compound is take part in an interaction with A chain and B chain also. This yellow color belongs to the B chain, and this light pink colors belong to the chain A. So after that, we go for molecular dynamic simulation. In this molecular dynamic simulation, we select the 8 compound, which is having binding energy more than 7. So for that, we use 100 nanosecond MD simulation. If you see in this movie, this selected compound is near to the basic spine region and slowly is move toward the surface of the CTLD domain, which is this surface domain is responsible for interaction with CTLD domain. If ligand is binding this side, so that will be inhibit the interaction with oxidized LDL. Then after we check that RMSD and RMS value. In RMSD value, we check that one, the stability of the protein ligand complex. If you see throughout this 100 nanosecond simulation, all compounds show this stable conformation. But just only if you see this purple color line, this is NPC 722, which forms more RMSD. And after that, we check the RMF value. We show that if you see, each amino acid taking part in interaction and how much they are going to fluctuate, they are showing through that figure. If you see this region, amino acid number 200 to 242 and 340 to 380, these amino acids are involved in the presence in the basic spine region, which is responsible for interaction with protein. So after that, we go for calculate the MMP-BSF binding free energy, which include that van der Waals, electrostatic, polar, non-polar energy contribution. We calculate the MMP-BSA for selected 8 compounds. If you see this red color compound, is this compound which is showing higher binding energy because they are going to morely interact with protein and involved in the formation of hydrogen and hydrophobic bond. Among that 8 compound, this green color compound shows the very least amount of binding free energy. So throughout this study, we are using this Mabridge and NAPS database, which contain 84,000 compound, which then we use this much compound for physiochemical property. Then we screen down 52,000 compound, which is used for 2D screening similarity. And through that 2D similarity, we screen down 30,000 compound, which is used for pharmacophore modeling. Then we screen down the compound, we get the 1963 compound, which is used for paints and molecular docking. Through after molecular docking, we select the compound which is having binding energy more than 7, which is contain 49 compound. from this 49 compound, we perform the clustering. We select the 2 cluster, and each cluster contain 4 compound, which is we select 2 compounds, means 8 compound will be there, used for MD simulation and free energy calculation. After that, we use this 8 compound. We select this 8. We screen this 8 compound throughout the 84,000 using this screening approach. This selected 8 compounds further we are going to use for biochemical assay and cellular assay to check the effect of LOX1 and oxidase interaction. So Mike Harris is my co-supervisor and Srinivasan is my co-supervisor. So thank you.
Samantha Hova.
Fabulous. Right. So I'm going to be talking to you guys today about essentially how we determine the structure of a viral protein, focusing more on the how rather than what the results kind of actually show. So as I'm sure none of you need reminding about viruses at the moment, but I work on a virus called Bunyamwera virus, and essentially viruses require your host cells in order to survive and replicate. And because they require host cells, there's sort of 2 main strategies for like sort of antiviral treatments. And one is targeting sort of host cell components that the virus requires to survive. And another is to target the viral components. So this can often require a structural understanding of the different viral proteins and how they're made up. Cool, right, so I work on the viral glycoproteins. And these glycoproteins are essentially cover the virus surface, and as such they're a major antigenic target, which I'm sure none of you need reminding about the SARS-2 virus glycoprotein, which is quite big, and there were lots of structures about that. And this protein is sort of main facilitator of virus entry into the cell, into like sort of the host cells, which can be one of the biggest barriers for a virus actually having a successful infection is the actual entry process. And through this, the glycoproteins actually go through a large array of structural changes. So determining this glycoprotein structure can help us with sort of vaccine and antiviral developments. But how do we actually do this? So Yehuda gave a lovely introduction to cryo-EM, so thankfully I can skip over some of that. And essentially what I just want to summarize by saying is that in these cryo conditions, we're trying to preserve our samples in a close to native state. And from this, you get a 2D production. So where you have your virus, for example, suspended in this layer of vitreous ice, and you hope your virus is in lots of different conformations, then you can use single particle analysis to computationally and align thousands of particles together and reconstruct a 3D model of what this looks like. But this relies on your particles, in this case, a virus particle being identical. And pleomorphism prevents you averaging and aligning all these particles together because they won't look the same. So what do we do if our viruses aren't identical, as was my case with my virus? So rather than say, for example, you've got your symmetric virus, you can see on the left here, we've got an example of a non-symmetric virus where the glycoproteins seem to be arranged a lot more randomly on the virus surface. So to do this, we turn to what we call cryo-electron tomography, where we 3D reconstruct individual virus particles rather than making an average of thousands of different particles. So to do this, the principle is very much similar in the sense that you fire electrons through your sample to get a projection image, but we do this lots of different times at lots of different angles. And here you can see some viruses suspended within a layer of vitreous ice, and we get this very lovely wobbly tilt series. And that's where sort of our first computing stage comes in, where we need to reconstruct this as a 3D image. And in general, this kind of applies the same algorithms, the same principles as other tomographic 3D reconstructions, for example, a CT scan, where we use this in combination with our cryo conditions, preserving our sample as native as we can. And the great thing about this is there's lots of sort of open source programs available there for you, which apply these algorithms to your tilt series, and there's lots of like guides and things available. And despite this sort of program that I use, iMod, being quite old now, it came out in 1996 originally, it's continually being updated, and there's new programs constantly coming out to make this easy, or relatively easy. And again, it's quite an old school looking, but it's very, very simple, simple GUI, and uses parallel processing to help reconstruct our tomograms. And here you can see an example of our viruses suspended in ice and the tomogram we get at the end, which obviously looks a lot better than our shaky tilt series that we began with. And that's all very good, but that gives us a nice image of our virus, but it doesn't really give us very much detail on what our glycoproteins actually look like. So to do this, we then do another process called subtomogram averaging, where where we align and average thousands of different subtomograms or subvolumes. So rather than in single particle analysis, where you'd say average the whole virus particle, you select your individual glycoproteins from the tomograms, and you select thousands of these, similar to what we do with single particle, and then you average these together, and then you get basically your glycoprotein structure. So on the right here, you can see an example of what your subtomogram average looks like, and then sort of a 3D rendering of that. So that's essentially the principle behind it. And I use a program called PEAT, or Particle Estimation for Electron Tomography, which again, it's an open source software which will help align all your particles together, and brings all the algorithms together to help you do this. And you start by manually picking a load of particles, so things that you think are a glycoprotein spike, to help give it an idea of what you're looking for. And we start off with this very blurry kind of spike sticking on top of a virus membrane. So this is about 500 particles. We then take our reference and then we apply it to all of our different tomograms, where we begin with initial model being, say, automatically selecting where you roughly think all the glycoproteins are. And then you use the reference to help you align all these to the reference and together. So initially we started off with about 30,000 particles. But obviously these aren't well aligned together. This looks terrible. And then this is where we go through an iterative process of refining both the positions in respect to one another and both the rotations, because they're not all going to be in the same orientation, to continually improve our structure. And again, this is a very sort of basic GUI where you give it your tomograms or your volumes, your models, and then you define a number of different rotations you want it to search through to find the best angle or the best position. And that's essentially what we do. So we start off with our model, and you get your initial average with its associated initial reference. You then put this reference into your next round of refinements. You then get a slightly improved average and a better reference, and then you put that into the next round, and you continually repeat this until you get sort of a more refined average, and essentially until you can't make it any better. So you keep going, and yeah, it's really refine it. And this, for our glycoprotein, is what we ended up with. So on the left, you can see an example of the electron density at the end. So the density itself is in black, and then just an isosurface sort of 3D rendering of what our spike looks like. So the spike you can see in yellow, and it forms this kind of unusual tripod-like structure. But this is still pretty low resolution for electron microscopy, and there were no published structures available. So for us, we're quite limited in terms of what we can actually do with this on its own. So this is where we turn to AlphaFold. So AlphaFold is a sort of product of Alphabet and DeepMind. So it's an AI-driven learning algorithm. It's based on the evolutionary changes in amino acid structure and sort of the physical constraints of how a protein is made up, so that you can basically give it an amino acid structure and it should predict a sort of 3D structure of your protein. I will say it's not always successful, but yeah, so for what we did, we were actually quite surprised with what it kicked out. So we've got two glycoproteins here, GN and GC, which form a complex. And we were quite surprised that these actually form something quite plausible. So then we fitted this into our electron density, and we could see that as a monomer, it actually fitted really well. And we can fit this as a trimer and see how this trimeric arrangement of the glycoprotein comes together. And it just kind of showed that we needed both the subtomogram averaging and the AlphaFold to predict how these glycoproteins are arranged. And then we can start to do some more fun things with this. So, I mean, we can map our subtomograms back onto the virus and see how it forms this sort of locally ordered, like lattice-like arrangement of the glycoproteins together. And then we can sort of build a bit of a bigger model with the AlphaFold model, and you can see how different areas of the different proteins come together. And sort of the take home really from this is that we can see the overall structure of the glycoproteins, which is great for us. And importantly, you can see things like the exposed epitopes. So the epitopes which actually might be available to a vaccine or, to your antibodies in a vaccine, or things like drug design. There's no point designing a drug to an epitope that's going to be buried, but it's something that you might not necessarily know without having this kind of model. I've managed to go over that very quickly, but we're done. But yeah, and with that, I'd just like to acknowledge everybody from the lab, our collaborators at Institut Pasteur, and thank you all for listening.
What's your honors that can still stick with 1 or 2 questions, so please. Was it the whole protein or just a pharmacophore? No, I know it's realistically domain. No, not in the document, just not in the document, just around the or using yeah, yeah. Okay. Questions, please. As our future records are, more coffee upstairs and then come right back for the lightning talk session. We can chat with the speakers as well. Another round of applause.
Speak here, I'll put this microphone on here. That's easier. That's preferred, yeah. You'll stand here. You can either have a clicker or you can navigate with the keyboard. It's up to you.
I've only got one side.
That's true. Yeah. I'm just thinking in general. Hi, Jenny, can you hear me?
Yes, I can.
Right, sorry, Jenny.
Can you hear me?
Super, hi.
Hello.
Okay, right, I'm just going to introduce the session and then we're going to hand over to you, okay?
Yeah, sure.
Super. No, that's not what I wanted. Right, Jenny, sorry. For the, if you could share your screen, just have the slide ready to go.
Yes. Share the screen.
Fabulous. Okay, super. I'm just going to introduce the session and I'll hand back to you, okay?
Yeah.
Okay. Good afternoon, everyone, and thanks for bearing with us. We're going to jump straight in to our combination of the poster session, which is a 2-minute presentation with a single slide, and our lightning talks, which are 2 slides at 5 minutes. So it's going to be short, sharp, and snappy with some excellent work from all sorts of people. So to kick us off, we've got Dr. Jenny Lee, who's going to talk to us about global sensitivity of an agent-based model. So over to you, Jenny.
Good afternoon, everyone. I'm a research fellow working on this project called BestMap, and my specialty is agent-based modeling. So in this project, I design and develop this agent-based model using NetLogo, and I'm going to introduce the tools that I used in this project. Hopefully this is helpful for people who are doing something similar. So first, the BestMap model, ABM model, is developed in NetLogo, which is a very easy tool to start if you're interested in agent-based modeling. In our project, we model farmers' decision-making process in adoptions of agri-environment schemes. And the aim is to assess agriculture policy, whether it's efficient, whether the design is good enough to get people on board. And in a model development cycle, we usually go through design, implementation, verification, and calibration. So in the later stage in verification and calibration, we need to run a lot of simulations to assess the model. And I'm using this R package, RX, which can run the model through R. And I'm using, it comes with embedded tools for sensitivity analysis and calibration. I have used this tool with HPC to carry out a global sensitivity analysis for this model. This method is called the Morris screening method, and I have listed all the references here. If you are interested, please have a look. And I think that's all for my 2 minutes talk. Thank you.
Super. Thank you very much. Next up, we've got Jai. Come to the stage and we'll just swap you around. Excuse me. This is your slide, isn't it? When you've got too many windows open. Is that good with you? Super. On you go.
My project is based on building a data pipeline to analyze symmetries in the network. A wide range of phenomena can be analyzed with the dynamic processes on network. For small scale networks, we can exactly predict the possibilities and exactly make the analysis calculation. For larger scale, we use the method of redundancy. What is redundancy? It is like reducing the dimensions for a larger scale network. How do we reduce it? By using the symmetries. Symmetries are nothing but a method where the where we get the permutation and combinations of different networks of combinations of a graph without affecting its structure. And why we have to analyze this network? Because the network can be by analyzing these networks, there are applications for the network is the voting behaviors of people, the way that people can influence the voting behavior on each other, or widespread of the disease during the epidemic. For example, for small scale network, I investigated for a small scale network of 11 vertices, which has over 1 billion data. If we there is a possibility for more than 100 vertices or million vertices, but as we increase the graph and as we increase the number of vertices, the permutation combinations for those will increase exponentially. So it will be tough for us to analyze those data. So we use the method of symmetries. For understanding the 11 vertices, I used HPC provided by our university. Thank you. I'd like to thank my professor, Dr. Jonathan, for his wonderful support and making me learn this new topic. Thank you.
Thank you, Jai. Just stop this now. Right. On my list, I have Eric up next. Yes, sir.
Waiting to come in. Sure. Hello, Monier.
Okay, where you go?
Okay, so I've just started a fellowship with the Leeds Institute for Teaching Excellence to apply data mining and text analytics and AI to decolonizing the curriculum. This is a major initiative at Leeds University. They're pouring millions of pounds into it, taking academics away from teaching and research to do special projects like this. And if any of you are teaching, not many teachers here, you'll know that when you do a module review, amongst the things you have to do is to say, how have you applied decolonizing the curriculum in your teaching this year? So I want to apply data analytics to this. So I applied the data mining methodology, CRISPM, the cross-industry standard process for data mining, that obviously all of you should be aware of. It's an example of a technology that you should be using in data analytics. The first thing is what's called business understanding, working out what it is the customer wants and what does this mean in terms of data mining and data analysis. So ideally, we want to find evidence of colonial bias in the teaching. Because if you want to measure it, if you want to decolonize as a verb, that means you want to change from colonial teaching to decolonial teaching. How do you measure things in teaching? Well, the data we directly have, at least in the library, is to have a reading list for each module. The reading list, the recommended reading for the students. We can't have, so that's, if you like, some concrete data that we can actually get at is the list of books and the authors of those books. Ideally, we don't just want the data, but we have to have it annotated, because if we want to come up with a classifier for predicting yes or no, is it colonial or is it decolonized, then you have to have some data where there are labels attached for each reading list item, for each book that's recommended. Is it decolonized or not decolonized? Or maybe is it colonial or not colonial? The problem is we don't have this. So the data collection phase of any data project we're stumbling at because we can't get hold of any realistic data. Potentially I might be able to get hold of data from surveys or feedback from students, but we're not allowed to access that. What we can do instead is measure the author of each reading list item. So the library and the IT service have put a lot of effort into tools for measuring or for extracting for each reading list item the author's name and which university they're affiliated with and where that is. So you've got a country code for each reading list item. And after a lot of effort and expense, they come up with conclusions. For example, most engineering and science teaching textbooks are written by authors who are affiliated with UK or US universities. So that's all that's as far as we've got so far. In theory, decolonization is about removing bias, because if, like, you know, for machine learning, if you have bias in the data, then you'll learn the bias. And in the same way, perhaps students are learning biased notions from this. In practice, we haven't actually got any data to verify or not verify this. But the library is planning ahead to add this complicated system to their toolkit, so that in future you will be able to see not just what you're recommended to read, but which country it came from. As to whether or not that does any decolonizing, that's up to you to decide. But at least it's got me out of teaching for 2 days a week to do this research for the next 2 years. And if you want any more information, I did a presentation of this at the Leeds Hi-Fi Comedy Club, along with the Turing Research Institute and the Cabaret of Dangerous Ideas a few weeks ago. It will be appearing on YouTube if you want to see the real funny act. Okay.
Thank you. Excellent. Thank you very much, Eric. Right, next up, we've got Stephen Slackhouse. And now we're moving on to our 5-minute lightning talks. Over to Steve. Oh. Red one's your pointer. Right, there we go.
Okay, so I'm going to try and talk a little bit about how electronic structure, so something very small, can influence something very big like the structure of the mantle. So to put things into context, I have a figure here showing the interior of the Earth. So at the center we have the inner core, and then we have the outer core, and then on top of that we have the mantle. So there's lots of exciting things going on in the mantle. So in green we have subducting slabs, we have thermochemical layers at the core mantle boundary. I'm not going to talk about any of that. I'm going to talk about the boring part of the mantle, which is the white stuff, which is normally just called normal mantle. So if you look at the right-hand side, I have a figure showing how the mineralogy of the mantle changes as a function of depth. So you can see you go through a series of phase transitions in the transition zone, as the name implies. And then in most of the mantle, we have 3 minerals: ferropericlase, calcium silicate perovskite, and magnesium silicate perovskite. So I'm going to focus on ferropericlase. So ferropericlase has the cubic rock salt structure. So it's basically magnesium oxide with about between 10 and 20% of the magnesium ions replaced by iron ions. And iron is a transition metal. And so when it's in ferropericlase, there's crystal field splitting. So you get a splitting of the d orbitals of the electrons. And at low pressures, the electrons could be pictured like this. So you have 4 unpaired electrons. But as you go under pressure, as you go down in the mantle, the increasing pressure causes the crystal field splitting to get larger, which means that at high pressures, it's more favorable for the electrons to pair. And so we get what's known as a low spin configuration like this. So why is this important? So one thing is, because these orbitals become unoccupied, if you imagine these are like balloons, you pop some of the balloons, the effective volume of the ion changes. So you get a change in density, and you also get a change in elasticity. And this is important because everything we understand about the mantle comes from looking at velocities, and both density and elasticity appear in the equation for velocity. It also affects buoyancy. So if you can imagine, you've got a process that's affecting the density of material, of upwellings and downwellings, that's going to affect, a change in density is going to affect its buoyancy, and it also affects its viscosity, which has implications for mantle dynamics. So people have been looking at this for a while. So there have been some really great studies by the Venskovich group and Wu et al. And they're trying to map out what will be the spin state of iron within the lower mantle. So we have a temperature here and pressure here. And these were great studies, but we found that didn't get such a great agreement with experiment. They found quite a narrow spin transition region, which you can see here. And so the elastic anomaly is narrower and it's deeper than we might expect.
And one of the approximations that they make, that there's an ideal mixing of high and low spin ions on ion sites. And a later study showed that actually there's a favorable enthalpy of mixing between high and low spin ions. And this makes perfect sense because you have a high spin ion here and here, they're both large. If one goes low spin and it goes to a smaller volume, then it means that this ion here has a more volume to remain high spin for longer. This meant that they would get a much broader spin transition. And in this figure here, we're just showing in the green dashed line is the results of Venskovich, which get the narrow transition, and then the blue line is the results of Holmstrom and Sticktree, which get a very broad transition. And the experimental results seem to have a slightly different gradient. And so one other thing about both of these is they only looked at one arrangement of ion, and their arrangements of ion that looked at were quite symmetric. So we decided to look at a multitude of arrangements of ion and look at random arrangements of ion. And we found two, I guess, N members. One is where you have two ion atoms which are on axis nearest neighbors, which you have in this case. And if that was the case, we'd find that one of those would always be the first to undergo the spin transition, and the other one would be the last. Okay, again, because as one goes low spin, it creates this volume to allow the other one to remain high spin for longer. If we didn't have any on-axis nearest neighbors, then we get a sharp transition like they saw in the other studies. Okay, so in the end, we came up with our results. We did 20 different configurations, and the solid red line is showing our results, which gives a much better agreement with experiment, which are the circles, than the previous approximation where you get you assumed the ideal mixing of high and low spin ion.
Right. take it away. Good to go.
Hi, my name is Thomas Hancock, and I'm from the Institute for Transport Studies, which is a relatively small department within the School of Environments. And I'm going to be talking about faster estimation of choice models. So I'm in the choice modeling research group, which basically means we observe behavior, human behavior, typically choices, and try to understand those choices and predict those choices. So what we might have, for example, within the transportation domain at least, would be sort of like travel modes choice, destination choice, things like that. And our type of data we work with can vary quite significantly. We could have stated preference data or real world behavior. So with stated preference data, the idea would be that we'd be looking at what would be the impact of a proposed new policy, for example, or just to try and understand how people would make decisions in a hypothetical scenario to understand what their sort of true preferences are. So I've got an example in the bottom right here where we've got example choice tasks. So in this example choice task, where it's probably quite small at the back, what we've got here is two alternatives. One is you stay with your current job, and the other is that you move to a new job where your commuting time is going to be longer, but you're going to get an increase in salary. So it's understanding how you trade off these different attributes. At the top, we've got an example of a revealed preference choice. So this is different possible routes someone could have taken for a journey in London. So they might walk, they might cycle. And what we have there is we can get sort of GPS data or a smartphone tracking device type thing. So we get sort of data there and look at sort of the real choices people have made, compared that to the other options they could have used, and then work out how they've made those choices. So we use a mixture of econometric and psychological theory to explain these choices. Basically, what we're doing is we're building mathematical representations of the decision-making process. We'll use these models to generate probabilities that a decision maker will make the particular choices that they've made. And then we're going to try and optimize our model subject to some parameters. So basically, what the real sort of ultimate goal of this is to put prices on something that doesn't have an explicit price. So we call this willingness to pay. So basically, putting a value on certain goods, for example, time. So the really classic example that's been around recently is the HS2. How much time are people, well, how much are people going to be willing to spend to save some time? So if you can say it's going to take 30 minutes less to travel from Leeds to London, how much extra will people be willing to spend to save that 30 minutes? So we're trying to understand these trade-offs, basically, rather than make a direct evaluation, because if you say very directly to someone, how much will you spend on this factor, there's going to be all sorts of hypothetical biases. Whereas if you do trade-offs, then you can try to understand how the different attributes of things will relate to each other. And the ultimate goal here is, as I've said, to predict future behavior, and we can then forecast scenarios to inform policy, for example, HS2. So what does this actually look like? So we've got, basically, we're going to have some big data set of choices. We're going to try and use a model to predict all of those choices in our data set. We're going to maximize the likelihood of the model. So quite sort of simple statistically. And what I've got here is just an example of how we've done this using the HPC. So I'm using the Apollo choice modeling package in R, and it has a nice little feature where you set the number of cores that you're going to use in the model. So the number of cores that it will use. So if I was using just my desktop, I'd probably want to use one less than what it's got so that I can do other stuff at the same time. So I might use three of the 4 cores for this model. And in this example case, what we've got is 3,500 mode choice tasks. We're using a model where we've got random parameters. We've got 1,000 draws for those random parameters. The model takes 59 iterations to converge. And this is for 15 parameters. And this means that there's roughly 6 billion probability calculations in this model. On my desktop, this model takes about an hour. On the HPC, when we use it to go up to 40 cores, it's down to just a few minutes. Now, to give an example of how useful this is for our work, in my most recent paper that I've been working on, we had 4 very complex models which each took pretty much the full time, sort of 48 hours on HPC. This is sort of roughly 10 times faster. So what I did is I set those 4 models to go on Friday night, come back on Monday morning, and I've got these 4 model results. But had I done that on my own computer, these models would have each taken 2 to 3 weeks, and I couldn't run simultaneously, so it would have taken about 2 months. Instead, it's done in a weekend on HPC.
Thank you very much. Next up, we have Yuan Chu. No, yours is this one. This is yours.
Yeah, that's
Good. I'll just check. Right.
Okay. Hi, good afternoon, everyone. My name is Sean. I come from the School of Chemistry. Today, I'm going to talk about the computational study of copper tungsten. Okay. As an energy production diagram shift from the fossil fuel to the carbon-free, low-cost energy, using the copper constant to split the water molecule into hydrogen and oxygen has become one of the promising ways to get the renewable energy. So in this work, we use the density functional theory for studying the electronic property of the copper tungsten. And here are some computational details here. We use the PBE function with the DFT plus U method to treat the exchange correlation functions. And we use a DFT-D3 with Ben-Johnson damping method to modeling the long-range van der Waals force. And here is the lattice parameter of the copper tungsten and its structure shown in the table and the figure. We can see that all the calculated lattice parameters are close to the experimental result within the 1.5% deviation, and also the oxygen atom are at four non-equivalent position, forms an approximately hexagonal close packing with cation occupying the half of the octahedral sites. And because of the Jahn-Taylor effect, each metal atom is surrounded by six oxygen atoms, forming approximately MO6 octahedral together. And we also plot the density of states of this of copper tungsten. The band gap is 2.24 eV, which represents it is suitable for the photocatalyst to split the water, and the valence band is composed by the O2p orbital, and whereas the conduction band is composed by the copper 3d orbital. Thank you, guys. That's all of my presentation. The next, the further work will be based on this book, and we will modeling the surface and do some redox property and also the water molecule absorption reaction on it.
Next up, we have Tamara. There's a clicker if you want to. Thanks very much. On you go.
Oops. So I'm Tamara James from the Centre for Environmental Modelling and Computation, and I'm going to take a quick break from high performance computing to talk about a bit of data visualization. So if you're an R user, you'll know about Shiny apps, which are a great way to build interactive data visualizations, and your users can modify inputs and see how the results change. So the standard structure for a Shiny app is that it has a UI component, and that takes care of all the front end stuff. and allows you to add inputs that your users can use to interact with the data and define the outputs. So in this example, it's a histogram of results of eruption times, and you can modify the number of bins within the histogram. And this works, the inputs get sent to a server component, which does all the back-end processing to manipulate your data and produce the plot that then gets sent forward to the front end. So this is a really nice framework, and it's really useful for standard inputs like plots and tables. But what if you want to visualize something a bit more complicated or non-standard than that? So this is the situation that I had working with some researchers looking at a literature review of climate mitigation studies, and they wanted to present the results in a visually appealing way that allowed users to manipulate the data and drill down into different categories and produce their own sort of outputs for use, maybe if they're policymakers to use to display in slides themselves. So you can do this in Shiny with custom outputs, and it's a similar framework to what we saw in the simpler example. So again, in the user interface component, there's a bunch of inputs, form controls for manipulating the data, and then there's an output element. And in this case, I've used HTML and CSS and JavaScript to produce the outputs. So the output element within the user interface sets up the framework for that. And then on the server side, there's a custom function to render the results, to produce the results and render them into an appropriate form to send through to the front end. So the bit of magic that is then required is some JavaScript. So a JavaScript binding which allows you to take the results from the data manipulation and output them into this HTML framework. And so this is a JavaScript object where you tell it where to put this data on the page with a find function, and then you give it a way to render the value. So in my case, it goes through the data frame and outputs table rows and then puts in the JavaScript to do all the fancy hiding and showing. And finally, you have to register that binding with the application, and magically it all works with your own customized output. So that's all. Thanks for listening.
That's my experience of JavaScript too. You just give it stuff and it does magic stuff. So I'm glad I'm not the only one. Super. Yes, we've actually got some people who are going to demo some Shiny apps rather bravely, I think, tomorrow. So it's excellent that you've showcased the framework here. Fantastic. Okay, so now we're on to the last two talks, which are Zoom talks. So Konstantinos, are you there?
Yes, hello.
Hello, super. If you want to share your screen and you can get started, I'm gonna start the timer and I'll give you a 30-second warning towards the end. Does that sound okay?
Okay.
Lovely, we can see your screen. So on you go.
Okay, good afternoon, everyone. My name is Konstantinos Biris, and today I'm going to present to you as fast as possible a new online application architecture. This architecture was explored by creating a mobile multiplayer chess game for Android devices using the Kotlin programming language. This talk is based on my FYP research project on ways to develop an architecture that would help reduce the cost of operation for online app developers. The first architecture that, the first architecture that came into being was the P2P lockstep. This decentralized model allowed users to connect using mesh topology. Each user runs the program locally on their own machine, and once a move has been made, a message is sent to all the other devices containing the move command, and each device executes it locally. Then, the client-server model made things easier, as the code would only run on the server, and the client devices acted as terminals, sending inputs to the server. This made the game more centralized and ensured that it had the same state for all users, which used to be a big problem with P2P lockstep, as having every device running the code independently could lead to desynchronization. Nevertheless, even this architecture had some latency problems. So currently, the most widespread innovation is the client-side prediction architecture. Here, the client does not act as a terminal. It executes the move locally and simultaneously sends it to the server, where it also gets executed. Then the server sends its game state to the client, where the two versions are compared. And if the client state is different, it gets corrected to match the one of the server. An example of this can be found in online shooter games, where you might see your avatar moving, and then it suddenly gets teleported to a slightly different location. That's your device correcting its false prediction after receiving the server's actual game state. So now the presented model combines elements from both P2P lockstep and client-side prediction. The game runs locally on its device, and the server plays the role of the mediator between the client. The client sends the two strings to the server. The first one is the move executed, and the second one is its game state. The server sends the move string to all the other clients, and then they run it locally. If there's a mismatch between the game state strings of the other clients, then the referee call is made. The server checks if the overwhelming majority of devices have reached a common state and democratically accepts it as the correct one. Otherwise, the server executes the move on its own based on the last game state it had stored and commands the client to adopt the resulting game state as the correct one. Here's an example of the architecture in action. In an online match of chess, a user reaches checkmate. The server gets the performed move and sends it to the other user's device. The two game states are identical, and thus the game culminates. So to test the architecture, a mobile online game of chess was created, and 50 matches were carried out initially. Then a tournament was conducted. During this session, 8 individuals from different real-world locations took part. 3 of them were in Leeds, 1 in Cardiff, and the other 4 in Athens. This method of evaluation was chosen because it resembled the potential real-life use of the app. The evaluation of the proposed architecture in terms of efficiency was done using manual and unit testing, as well as Firebase's analytics feature. It was indeed proven that after conducting almost 60 matches of chess, the amount of data stored in the Firebase real-time database was kept at a minuscule size of 13.32 kilobytes without deleting any of the entries. Not only that, but the time it took for the app to update the game state fell between 0.5 and 1.5 seconds, with the average time being equal to 1.1 seconds. Finally, it should be noted that although this architecture might be beneficial for more slow-paced tasks, it has not yet been proven to work in the same way for more fast-paced functions, like those required in online racing games or video conferencing apps, etc. It remains to be seen. That's all from me. Thank you all very much for your attention.
Thanks very much. Super, great. Okay. And that, if you want to stop sharing, Konstantinos. And next, our final speaker is Munir. Hi, Munir, can you hear us?
Yes, hello.
Hello. If you want to share your screen, and we'll get started.
Okay. Can you see my screen? Okay.
We can. Fantastic. I'm going to give you a 30-second warning, but the floor is yours.
Hello, everyone. I am Munir Al-Aidi, a lecturer at Princess Noura bin Abdulrahman University in Riyadh in Saudi Arabia, and a PhD candidate at the University of Leeds in the School of Computing. I'm happy today to participate in this talk and present my work in TK and SBERT and their use in parallel sentence alignment using transformers. A parallel synthesis corpus is an essential resource for many applications of artificial intelligence and natural language processing, including chatbot, which is the main research problem in my PhD research, along with other systems like translation, question answering systems. And by improving the quality of the parallel synthesis corpus, the performance of these applications is improved. In my work, I study first the use of the AI and NLP technique to transform a large number of English documents and their Arabic translations into a set of aligned parallel sentences. The data set I use in my work is the Saudi Learner Translator Corpus, which is a corpus that provided by the language college at Princess Noura University, which is a bilingual corpus from English to Arabic. And it's contained more than 366 different files in different contexts. In my work, I choose only 109 files in the healthcare context. I start my work in data preparation and processing, pre-processing in the document level. I work to align each English document with its corresponding Arabic document. Then after this step, I start the sentences tokenization using the sent tokenize, the NLTK sentence tokenizer tool to convert all text in the corpus into sentences with different lengths. During this process, I explored the data and found some problem inside it, like we found many unrelated information to the Arabic and English corpus, like student information, student ID, student name, and try to clean the data set from this information. I found also some missing files, like I found the source English text disappear and the corresponding Arabic translated text are missing, and also some wrong data entry switch between the Arabic and English text. So we end up from this stage with 88 document peers in the healthcare context. After that, we start the pre-trained, the modeling. I use the pre-trained model expert, where we apply for multi-language expert pre-trained model to match each English and Arabic sentences based on their highest consonant similarity score. And the output was like this table, a table with 3 columns. The first one have English sentences, and the other one is the aligned Arabic sentences. And the third column contain the cosine similarity between each sentences. The result we end up is we have more than 20,000 parallel sentences from the original 88 paired document. And in the table here, you can see the average cosine similarity score for each pre-trained model. And I try to analyze the results I have, and I think that some of the misalignment, the reasons for the misalignment is because the inconsistency in the number of sentences between the original English text and the Arabic translated text, because some of the translators try to shorten in the text or translate the text in general and not identical to the original text. Also,
30 seconds.
The inconsistency in the length of sentences, too short sentences or too long sentences. Thank you all. Here is my emails if you can give me any question or suggestion to improve my work. Thank you.
Thank you very much, Manir. Super. And that brings us to the end of this session. We have overrun slightly because we are slightly overrunning, unfortunately. So there unfortunately isn't going to be time for questions in this session, but what would I encourage you to do is, if you do have questions for anyone, feel free to catch them in the coffee breaks, or use the Slack as well as the other medium if you've signed up to the Slack. But unfortunately, we just don't have time for questions just now, because immediately we're going to start off our panel discussion in here, which will be on open research. So we're going to get our panelists up to do that. That said, if you're also interested, there is a code clinic happening now over in 849N, which is looking at the Conda package management system. So if you've ever heard of that or want to find out more, you can pop along to that. But we're going to take 5 minutes quickly just to change this room over for a panel discussion, but be back in here for all things open research. Thank you very much.
All right, welcome to our next session. This is going to be a panel discussion on open research. So thank you very much for joining us. I'm going to first introduce our panel members. Then I'm going to start with a question that the panel members can comment on, and then you're invited also to come in, comment, or answer the question as well. So we want to do it a bit interactively. We have overall like about 4 questions, but we may decide to just do 2, whatever. So my name is Victoria Spazer, and I am an associate professor here at the University of Leeds at the School of Politics and International Studies. So the first panel speaker is Kelly Lloyd. She's a PhD student at the Leeds Institute of Health Science and the lead organizer of the Responsibility Tea Journal Club. Thank you very much. Unfortunately, Marlene, who's also supposed to be here today with us, couldn't come because she has COVID. She has positive COVID today morning. So instead of Marlene, we have even 2 more speakers today that I could convince to participate in this panel. So it's Martin Callaghan. I'm sure you all know him. He's really he was the main responsible for HPC and ARC. So, but now he has moved to the School of Computing and is going to do his own research. So that's quite exciting. And then we have Graham Blythe from the Leeds Library, University of Leeds Library, and he is there responsible for research data management. Thank you very much for being here. So the first question is, what does open research mean in your discipline? And I'm going to start with you, Kelly. Okay. Thank you very much, Kelly. Martin, to you.
Great, thank you very much indeed. Well, thanks for that. And I don't think open research in computer science is probably very different in its total view. So I made a few notes about what I thought it was. So I think what you described there was a set of values and practices, really, that sum up what open research means to you. And I think all of what Kelly described can probably be applied to every scientific domain or every research domain. Because what you've really talked about there, I think, are sort of values and practices that make research easily discoverable. As academics, we want our research to be discovered because our research is our next job, isn't it, fundamentally? To make it transparent so that people know how we've arrived at our conclusions, and that they can see into the ethics behind the nature of research that we've done, Importantly for UK funders, they want to make sure that the publicly funded research we do and the publicly funded data that we produce is publicly available and available for others to use as well. And I think certainly in the UK that's become critically important. It's an investment, and open science underpins that investment. But I think more widely than that, it perhaps underpins a culture, an inclusive culture about collaboration and encouraging us to collaborate openly, not only with our colleagues in our schools and research institutes, but more widely in the sort of formal and informal research areas and research groups across the world, improves the quality of research that we do. So I think fundamentally for me, open research and open science, if we want to use that term as well, is about helping us to share more what we do, and by doing so, challenge and be challenged in our findings. So that's my outlook. So I'll pass over to Graham.
You're taking over chair's duties as well.
I could pass it back.
Just a comment first. Poor Marlene, who couldn't be here. Marlene is a researcher in mechanical engineering, biomechanics, and all this stuff with body parts. And Marlene couldn't be here, she had to be replaced by 2 men to make the same level of contribution, which is fair enough. Marlene's work, for example, is getting tissue from tissue banks. If we look at the whole life cycle, sometimes your research starts with stuff that isn't yours, a bit like yours, Eric. You build up corpora of material. You didn't create the Quran, as far as I know. So the whole life cycle, in Marlene's case, she gets material, she does experiments, she writes models, she develops code, all of which is shared in different ways. And the real question we get in our team from researchers like Marlene is, what should I share? What does represent open research? And our usual answer is, don't ask us. You are the experts in your disciplines. If you read a paper, what do you expect to find that would allow you to validate or verify those findings, to truly understand those findings? That is a sort of an academic end of what represents open research. And that's without even going into citizen science and public good. But I think that one question, what would be needed to support your research findings to allow someone else to truly understand your work? That's the start of open science.
Did you want to comment on this, Kelly?
Oh, no, I was just going to pass you this.
Sorry. Okay. Actually, good idea. Maybe I just want to give the public as well a chance to ask any questions that you may have, or maybe comment on your own practice of open research. Please, Eric, pass this on to you.
Okay. I was actually surprised by the first speaker who said you're a medical researcher and you're allowed to make your data open. Maybe I work in text analysis. And my experience has always been it's actually been quite difficult. For example, I had a supervisor PhD student who was working on text transcripts of medical intervention clinicians and stuff, and he was not allowed. He could collect the data for his PhD research and then throw it away at the end. He wasn't allowed to make it open access. I've examined PhD theses in text analytics at Sheffield, for example, where they've done medical text analysis research, and then had to lock up the data because they're not allowed to make it open. I've even had an EPSRC funded research project where it was co-sponsored by the Centre for Protection of National Infrastructure. It wasn't medical data, it was terrorist detection data. I'm not even allowed to tell you what the data was or how we got hold of it, let alone make it open access. So how do you square the circle that for medical research in particular, that you are allowed to make it open?
Thank you, Kelly. I actually would like to pass on this as well to Martin and Kalle again, because I think, Eric, you did raise an important issue, like the limitations of open research, and how do we deal with that?
So I think we've got probably two things to consider there. So of the particular set of research or experiments that you were considering there, we've got the data, which is one thing. And then there's your code, your algorithms, your analysis, which is another. And I think it's perfectly possible for one part of that to be open, but not the other. So if that data is subject to any form of agreement or even licensing, if it's commercial data, then it may well not be possible legally or ethically to make data available via any sort of open forum. But that doesn't necessarily mean that your code, your analysis, your algorithm isn't. And it can still have value to share your methodology in as much detail as you get from the code with other researchers who may well be able to use that on other research projects. And I think that's possibly an important consideration. It doesn't need to be all or nothing. We can look at the different stages and processes within our research process. Some of it may be appropriate to be open. Some of it may not. And I guess there are probably many of the things that you've discussed with research colleagues in the library, haven't you? That it isn't all or nothing.
Yeah, Martin, absolutely. And I was going to say very similar words. The one thing I would add to that is material like Kelly's doesn't just become open. It has to be planned for. So I'm assuming, Kelly, you went through ethical review. You probably had to go through NHS ethical review, maybe not, but it doesn't really matter. The point is plan for it.
I would just have to get consent forms. I got consent for distance, and if I hadn't done that, I wouldn't have been able to do it later. I had to plan for it.
Yeah, that's it exactly. If it's a contractual agreement with a company that you're collaborating with, get that agreement nailed down at the start of the project for sharing at the end. Don't wait until the end of the project to think about the openness of your data, your code, your methodologies. Plan for it.
Thank you very much. Eric, does that kind of address your issues, or would you like to come back to it, like with maybe more comments? No? Any other, any other questions from the public? Yes, Serge.
Okay, actually, my question is related to sharing code, and this is partly back to the past experience of Martin here. And there is for a story, right? I can illustrate this with a story. There was a PhD student in an obscure small university in the Czech Republic. And the kind of research, and this was in the beginning of the 2010s, right? And he was doing some kind of elaboration on existing on research which occurred towards in 2000s, and then he got his PhD in 2012, and then essentially what he did, he got a job at Google. And with this, he published the outcomes of the research in the form of a software package, which is called Word2Vec. And those who worked with natural language processing, they know, because it became an instant hit, right? And at the same time, so then during the process, while he was doing his PhD, very few people knew about him and about the kind of research he was doing. But then once this was packaged in the right way, with the help of software engineers at Google, it became a hit. And then this also applies. So then on the one hand, I do encourage my students to publish their code, and most of the time in natural language processing. So unless you work with terrorists, this is not an issue. The entire industry of natural language processing works on the basis that that code is publicly available. But then there is difference between what, for example, I produce or my students produce and what is produced by Google, and therefore this creates some kind of disparities. And then, so essentially, my question is about the role of software engineering in this process, and the research software engineers.
Thank you.
One shot, so yes.
Ja,
Thank you. Kelly, would you like to comment on that as well? Okay, I mean, I think you actually raised an important issue that was also on our list of questions, namely what actually makes really things open, right? I mean, what else do we need to publish along maybe like code and data to make sure that it is really reusable, that it's really replicable, and that's really understandable. So I think you mentioned some of the things that potentially you want to maybe like create like a image of your environment, like, you know, within which you kind of create your code, right? So like if you, for instance, work with Python, like, you know, it's becoming very likely that it becomes unusable if you don't also kind of save basically your environment within which you kind of create this code, but other things maybe as well. So maybe Kelly, you want to comment on this?
Yes, that's a really important point about not just thinking about it as data, but everything else. So, for example, with a survey study of mine, I'm currently looking to sort of put online. I started thinking, well, actually, what do I need apart from the data and the analysis script? And that got me thinking, oh, actually, if people really want to understand how I did my study and make it as open possible, I thought, oh, participant information sheets, consent forms, survey materials, everything I provide as the participants. So this can be replicated, or even just so someone can actually understand what I did. I feel like we might all have experience of reading a paper, and if there's no additional materials, because journals have incredibly short word counts, it can be really difficult to actually work out exactly what the person did. And I think yes, there's just so much more to be added on. And I have had experiences where I couldn't actually share the data because we hadn't got prior ethics. And it did make me think, oh, well, there is lots of other things I can share. I can share the questionnaire and I can share the analysis script, and I can be as open as possible within these restrictions I have. And there's no harm in it from my point of view. I don't see a harm in sort of sharing that survey or anything. So I just see it as a plus to try and make my paper as easy as read as possible. And hopefully it makes it easier for someone in the future as well. You don't have to redo what's already been done. We can make it a lot easier for the research community.
Thank you. Would you like to add to anything to that?
Well, I could add a little bit. And
Thank you. Are there any questions from the public? Yes, I think you are first. There are actually many.
Cheers. I was just wondering about both sharing outside of just code and just data, and helping your science be more reproducible. There's lots of ways to do this. For example, with coding, you could publish an open software paper or something. I was wondering if you could comment on the ways that you see research in the future helping to incentivize this, and in general academia helping to incentivize this. So for example, when it comes to research coding, incentivizing using software engineering techniques, incentivizing continuous integration, incentivizing us publishing software papers, because I can't comment from it like further on in this point of view, but as a PhD student, I've chosen to go that path despite the fact it eats a lot into my time. It's not really recognized in our discipline yet. And say, for example, in a thesis by publication, software papers are very much, you know, they're not considered enough of a contribution. And I was just wondering, what do you see in general as a good way of incentivizing and promoting, and where is the future going when it comes to incorporating these things into academia and as sort of like, yeah, in the next stage and incorporating open research?
Thank you. Please. Oh. Right, okay, then I'm just writing back. Who wants to go first?
Go on, I will. So I think you've asked a really good question, and in fact, you gave the answer, which is about incentivizing. How do we incentivize those practices? And I think most of us have probably worked out or already knew that academia is incredibly conservative, and it really, really doesn't like change. So I think part of our jobs is to challenge the status quo, is to start to say these things are acceptable. These are techniques that improve the quality of research. They improve the quality of research outputs. And it has begun to happen. So research software engineering, you mentioned, that did not exist 10 years ago. And now in many universities and in significant proportions, significant parts of the world, it's regarded as good practice, and there are people employed both to be research software engineers supporting science and research, and people like yourself who have said, well, actually, these techniques are really good because they help me with my research. And we've got to work together to challenge that status quo. So it's my responsibility and Graham's responsibility and Victoria's responsibility to sort of stand up for PhD students who are doing this and stand up for postdocs to say, no, this is right, this is appropriate, and this is how we make research better in the future. And we can definitely see, I think already, a track record of how this has improved the quality of research. You don't have to use all of these techniques all of the time. And you've probably noticed just by employing one or two of those techniques that there's a bit of a learning curve, but then the rest of your research process becomes so much easier, doesn't it? You save your time, so save yourself time by learning, by spending 3 weeks to learn a technique. You'll save yourself 6 months during the course of your PhD.
Yeah, I'm not. Incentives are always tricky, aren't they? We've got REF. This sort of open research, maybe REF will get better of that, and that will really help. But I think looking nearer to home, I think you are part of the solution as well. If at the start of our projects, even at the definition of the project by the PhD supervisor, as we're applying for a grant, we recognize materials and resources that are already out there, cite them and reuse them. We're using the built-in academic, if you like, system of recognition. That is citation. So if we share this stuff with DOIs, with some other identifier, and then when we reuse this material and we actively search for this material, rather than jumping straight to our keyboard to write our next bit of code, what else is out there that's pretty good? Let's cite it, let's develop it further, and hopefully we get a virtuous circle where everyone gets the recognition.
Thank you, Kelly.
Yes, it's a really interesting question, especially as I myself a PhD student, and I understand what you mean. It can be a lot more work when I'm trying to make sure I can put my data online and everything. I do sometimes think, wow, I've really added on to a lot of extra time, and I'm also trying to get my PhD done. I get that. I think there is a culture change that's happening really slowly, and I wish it happened faster. There's a lot of bottom-up pressure, and I can see it happening in places. I can see it happening from funders are changing some of their policies. There was a whole science and technology committee talk where people gave evidence on the problems with reproducible research in parliaments, and there are becoming these culture changes, and I have heard of open research practices coming into hiring criteria. So it is slowly getting there. So I guess I would just say culture change is really difficult, but I was going to say that you're ahead of the curve. I think the change is going in that direction. And I think those skills are becoming more and more needed in researchers. And I think it will benefit you in the long run that you learnt them how to do all that in your PhD, rather than the culture changing further along an academic career where it's a lot harder to learn these new skills.
Thank you. I think we had like 2 more questions from the public, and I'm going to take them both now because I actually want to make sure that we have a small break before the next session starts. So I think it was you.
So my question is related with paid tools. So for example, I write a code, I make the code publicly available, but it's written in MATLAB, for example. So how open research is allied to paid tools? We need to avoid as much as we can, or it's just doesn't matter?
Thank you. And then there was one more question from another member of audience. Oh, yeah. Sorry. sorry.
Kelly, right? Yeah. First of all, I'd like to congratulate you by your personal achievement and initiative for making your research open. And I'd like to, in life sciences, we have this, in my opinion, this disgraceful race for impact factor score. So, and I think it started 60, 70 years ago with the journals, the big journals, publishers, having copyright for the research they do. And this is the whole thing. People still want to hide something. If it passes through a couple of reviewers and an editor, it's okay. They didn't notice I hid something in my methods, and it's okay. And the paper is published. So it needs to come from us being open, and making it open. Myself as a reviewer, I always encourage. To me, it's a mandatory stuff for them the authors to make it open in a GitHub page, or all their codings, it must be publicly available. But sometimes if the editor doesn't have any computational skills, he or she doesn't mind and the paper goes through. So my question for you is, have you picked your journal? Are you in this race? I bet you want the best impact factor for your research. So I'm not sure about psychology, and I'm speaking for life sciences, folks. Yeah, that's my field. But and I'm sure if the reviewers are not interested in your coding to make it open, you will do that in your paper as well, right? So I'd like to hear your opinion on that.
Thank you. To go into you then.
Yeah, just to answer that question in particular, I guess with impact factors, obviously there's pressure, isn't there? I'm a PhD student and I want to go further in academia, so there's pressure to publish and there's pressure to publish in high up journals. I have tried before actually to publish looking at more sort of impact factor in high up journals, and that was quite long and I got rejected. Kind of more nowadays, I've had a really positive experience, for example, going for a journal that is still quite well received, but it also likes open research practices. So there's a research practice called registered reports. It's much more done in psychology, but it's also, this is one of the few medical journals offering it. And it's basically where I submit my protocol to a journal, and the peer reviewers look at it, and I'm granted in principle acceptance, basically based off the protocol. I then go away, do the study, and they come back to review it again at stage 2. And they basically confirm that I did the study how I said I would, and comment more on the discussion mostly. And basically takes the pressure off. I've got in principle acceptance. They can't really reject it unless I did a completely different study. And I was just thinking about how that had a really positive experience with that, by looking at a journal that kind of valued those open research practices a lot more. It's been quite positive. So it's just to say, I guess, yes, there is a lot of pressure there with impact factors, but I think I'm starting to look more and value journals more that do talk about open research and discuss those practices more, because it makes it a lot more positive experience when they sort of appreciate those sort of aspects. I'm not sure if that fully answers the question, but just to sort of go through how I've sort of changed my thinking throughout my PhD.
Thank you, Martin. Maybe to you, and maybe also the first question.
Yeah, I'll go for Patricia's questions. Patricia, just to recap, I think you basically asked, do proprietary tools, software like MATLAB affect open research? Really good question, because I think there are two answers to that: yes and no. There we go. The no, because you can still publish your code, you can publish your analysis, you can publish your results, you can publish your data. And in that sense, anyone who has access to that tool is able to reproduce and share in your findings. So in that sense, perhaps code that is published in MATLAB or something else, where there is a cost involved, a proprietary or closed source tool, is just as accessible, providing you have access to the tool. But maybe there's a no as well, because that's all well and good for those of us at universities and research institutes in the rich global north, where we can say, well, yes, well, every university in the UK has MATLAB. It's no problem. It's fine. Everybody can reproduce that. But that is certainly not the case in the not so affluent global south. And perhaps we do have a responsibility to think about research globally and not just in our own backyard. So my answer then is, well, actually, it makes a huge difference, because it means that my future collaborators in Africa or some countries in South America are going to be locked out of my research and research findings for no reason other than finance, which I think would be a retrograde step. So that's perhaps why the open science movement also is very allied to the open source software movement, and that's not really by accident. So I shall pass over to Graham. I'm sure we'll have some comments to add.
I would just echo what Martin has said. Although I would add to that, I think you're asking absolutely the right question. And I think it is a valid question for anyone to ask themselves. I would look at MATLAB and say, do you know what? It's plain text. It's human readable. I am sharing something that could be read and created in another tool. Now, if it wasn't MATLAB you'd used and it was something that you built within a GUI in something that generates some binary stuff that's really impossible to read without having the tool, I would push further to the left or the right. MATLAB, I see, is at the reasonably friendly end of sharing, but it's the right question to ask. And there are tools that are much less friendly in terms of their native formats and how open, accessible, even readable those are. Great question.
Thank you very much. I would like to thank the panelists for those great answers, and thank you very much as well for our public to have those questions, and to our audience for the questions. We're just going to have quickly time to swap over for the next session. Thank you very much.
Hi, Megan, can you hear us?
Yeah, I can hear you. Thank you.
Super. We actually have a withdrawal, so we're going to ask you to go first, if that's okay, but we're still setting up, so we'll give you the nod when it's time. Is that okay?
Yeah, perfect. Thank you.
Megan, please. She's going to present some work on connecting Bradford with APIs for automated data linkages at a district level. Megan, please.
Hi, everyone. So firstly, I'm sorry that I can't be there. I was there this morning, but I wasn't feeling very well. So I'm going to do that this remotely, if that's okay. Let me just share my screen. Okay, can you see the main screen or my note screen there? Sorry.
You can see a note screen.
Has that sorted it?
Yes, it has.
Perfect. Okay. So apologies for anyone who was at the leader event last week, and you may have already seen this, but hopefully that's not actually many of you. So again, just a couple of caveats. When I use the term API in this talk, I refer to it in a more general sense, rather than being more specific to web development. So just the automation via two connected programs, and most of the graphics that are presented in this talk are screenshots of live dashboards. So they're not actually interactive today, but they're just screencasts of our interactive dashboards. So over the course of our lifetime, we interact with many different services. So this can be health visitors as an infant, working our way through the education system, visiting our GP when we're sick, being the victim of crime, or being admitted to a care home when we're elderly. And each time we interact with one of these services, our data are routinely collected. However, these services often work in silos, and data from each of these organisations are not shared or connected, and nor do they often interact. So enter the Connected Bradford database. This is a pseudonymised linked database of over 600,000 citizens from across the Bradford district, and it allows us to connect data from multiple services into one place. So here we have individual level data from a range of services of organisations, as you can see in the diagram here. So for example, we have data from GP practices, from social care, from education, emergency services. So every time someone makes a 999 call or visits their GP or anything like that. We also have data from local councils as well. So lots of data all coming together for all the individuals of the Bradford district. And when we link these data, we can build cohorts of individuals that share particular characteristics and then answer specific research questions about these individuals. So working with researchers, with stakeholders, with policymakers, we can develop early interventions and even change policy to prevent adverse life outcomes related to health, well-being and social mobility. However, as I'm sure many of you know, data are messy, especially when they're routinely collected. They're not actually collected from researchers or academics, and data providers don't necessarily understand the importance or the impact of good quality data or the power that they actually hold. So to link the various data sets, it's vital that we standardise each of these, and we use the Observational Medical Outcomes Partnership, or OMOP, and their common data model as a sort of standard to comply to. So this, using OMOP's CDM, it transforms data sets into a common format. So they have shared terminology, vocabularies, and coding schemes, and this allows the disparate data sets to be linked and analysed coherently. So when we first started, we originally set out by manually writing our scripts to do things like parsing dates, omitting illegitimate values, etc. But this obviously takes a lot of time, especially when we have quite large data sets with many, many variables and many tables within these data sets. So this could often take days of work for an analyst to work through one of these data sets. So as a way around this, one of our senior research fellows, Sam Rellins, developed an API via a Jupyter notebook, and this allowed these processes to be automated. So we created a pre-processed data set ready to go for additional, more specific data cleaning within minutes and a few clicks from the analyst. So this meant that there was minimal human input, so we didn't have to write lines and lines of code. And so the risk for human error reduced drastically. And it meant that across the, all the different data sets, they were standardized according to a single model. So streamlining this process in this way has provided us more time to explore how we can best showcase this fantastic data that we hold to other researchers who may be interested in using it to answer particular questions. So during this time, we've created interactive dashboards within Data Studio from Google. And this allows us to view the demographics of a particular sample. So we have their age, gender split, geospatial location, ethnicity, etc. And this can be data from a single data set. So here we have the Bradford District Care Trust, which is a community care data set, or it could be multiple linked data sets, such as children's GP visit information and school absences. And these dashboards serve a dual purpose. So firstly, they allow us to sense check our data before they're released into the wild. So for example, here we can take a look at the demographics of a more specific cohort of individuals, so those diagnosed with asthma. And we can see how many individuals there are in the whole data set. So we have over 100,000 individuals who we have data on that we have, we have data that they've been diagnosed with asthma. And we can filter out these specific demographics at the top of the screen. So we can see how many individuals we have according to various demographics. So we could filter out for just individuals from Asian or Asian British backgrounds, males, females, age, etc. In addition, we can drill down into the more specific descriptives, which are specific to that particular data set. So here we have age of asthma diagnosis. So as far as our data are concerned, it seems that the majority of cases are diagnosed during early childhood, as you can see from the top right plot there. And I don't know how well you can see on the board, but the chart underneath would suggest that although there's fewer individuals from ethnic minority groups, the diagnoses do occur around the same sort of time, so the same patterns seem to occur regardless of the ethnic group. So these sense checks allow us to eyeball our data at a glance, see if they look right. So if there were no males, for example, at all, according to these visualizations, this would be a cause for further investigation. Okay, is this actually the case, or is there something actually wrong with these data sets that we need to fix? The second purpose of these interactive dashboards is that they allow researchers with specific research questions to explore the data that are available within Connected Bradford before they make a formal data request. So for example, if a researcher was interested in answering a research question that required data from 3 different Connected Bradford data sets, so maybe they wanted to look at GP visits, which are held in primary care, intensive care visits, which are held in secondary care, and health visitor information, which are held in the BDCT, which is community care. They could check the number of individuals that have data within all 3 of the data sets to check whether their analyses will actually be robust enough and have enough power. So here in the Venn diagram, we can see the overlap of individuals across these 3 data sets. So as you can see, primary and secondary care, the blue and green circles are the largest. So we have the most amount of individuals within these. So over 1 million citizens within these 2 data sets, while BDCT is a lot smaller in orange, but still quite substantial as far as samples go. So from this dashboard, a researcher would be able to determine that there's over 230,000 citizens with data in all 3 of these data sets. And by most people's standards, that's quite a mammoth sample size. So these are just 3 of the data sets within Connected Bradford, but obviously we could do this for all the different data sets that we have. So a researcher could see just how many individuals they want for their particular research question. So far, one application that we've used these data for is the exploration of various outcomes of children with autism. So here we have linked data from primary healthcare and education to investigate the achievement gap of children with autism. And as you can see from the figures here, children with autism in yellow are less likely to achieve 1 or more GCSEs at A to G compared to children without autism in blue. Indeed, as can be seen in the blue chart, children with autism are more likely to leave school with no GCSEs at all, whereas children without this diagnosis are more likely to achieve 9 GCSEs A to G. Currently, I'm looking at something similar, but a little bit different. So I'm looking at reproducing some previous research conducted, which explores relationships between a measure of children's school readiness and later academic achievement. So this work is currently ongoing, but it seems that children who are considered school ready according to the early years foundation stage are more likely to perform at expected levels or above expected compared to those who are not considered school ready. And this is the case across reading, writing, maths and science. So we hope the creation of these data sets and dashboards will enable ourselves and other researchers to answer crucial research questions like the ones that I've talked about today. And ultimately, we want to improve the life, health and well-being for the people across the Bradford district and beyond. Thank you.
Thank you, Megan. Thank you. So our next speaker is Payman Babakhani. He will present a model, model builds model, mimicking process-based numerical models using machine learning for Earth environment modeling. So this is a session that will not be live transmitted and recorded.
Do I change this slide?
So this one to progress, this one to go back.
Okay. And the laser beam
is that red one, yeah.
Good afternoon, everyone. My name is Wajiha, and I am a second-year PhD student at the School of Mathematics. And in this presentation, I want to share with you my journey of building a numerical wave tank. So I will start with the introduction of what wave tanks are. Maritime industry, they use these wave tanks to study the behavior of water waves and to study how to different models will interact with water waves, so it can help to build better structures for ships and for floating wind turbines and other things. But the challenge is that these kind of wave tanks are very expensive. One, to build, because you can see they're really, really big. So you need a capital cost. And second is manpower. The people who operate these tanks is also charge money. So to tackle with this problem, many researchers, they are developing numerical model of these wave tanks. So it's fall under the category of computational fluid dynamics. So you can solve a problem like this by using your computer. But the challenge is that these kind of models are very expensive when it comes to computation. So to deal with this problem, my research group, and we are working to develop a model which is based on potential flow. So it means it requires less computation power, and you can solve a problem like this by using your own laptop. You don't need a very high computing system to solve problem like this. So in this presentation, I will explain how to develop this model step by step from simple to more complex one. And I will also discuss the new implementation technique for the equations that we have developed that can automate the process of implementation. So the goal is to develop a numerical wave tank with wave flap wave maker. And to reach that goal, we are going through step by simple model and then add more complexity and gradually reach towards that goal. So I will explain each of them in each section of the presentation. Before I start explaining the mathematical and numerical model, I would like to introduce some of the terminologies that, if you know, it will help you to understand. So the model consider a rectangular wave tank, which has moving boundary on the left-hand side, a solid wall on the right-hand side. The bottom is fixed, so no slip condition, and the top surface of the fluid is free to move, so we call it free surface. And this is velocity potential. It's like a scalar, and it has some values at each point of the domain. So when you take the derivative in specific direction, you get the velocity in that direction. So first of all, I will explain this new method of implementing the equations, because then I will be using this implementation method in other models. So it's important to know either this new method is giving a result equal to the traditional approach or not. So let's say for this case, we assume a simple problem, linear shallow water equation, and the variational principle is given in equation 1. So you can see variational principle just as a conservation of energy of the system, because we are dealing with energy, how potential kinetic energy is changing. The traditional way is like we manually derive the equation by taking variations of the velocity potential and free surface to get these equations, which is 2 and 3. And then we implement these two equations in our numerical model to get the results. But what we suggest was that instead of deriving manually from step 1 to 2 and 3, why don't we just use the equation in step 4 and let the software compute the equations by itself? So it means it eradicates manual error and speed up the process. And then we did the comparison of a new approach with the traditional approach, and we found that the results are kind of same. It's the differences in the machine precision. As you can see, it's 10 raised to the power minus 13 and minus 16. So in our future models, we will be using the new method of implementing the equations. Okay, so we add the complexity to our linear model by adding a piston wave maker. So now it has a moving boundary, but still, because it's a linear system, so the length of the domain is not changing and is still stationary. You can consider it like this. The variational principle in this problem will look something like in equation 5, and the motion of the piston wave maker is written in equation 6. The time discrete variational principle, which is our novel way, is mentioned in equation 7.
So sorry, what's n, or is that something that you're solving for as well?
Yeah, so n is showing the time discretization. N is current time step, and n plus 1 is future time step. And this discretization is based on a method known as symplectic Euler. And the speciality of this method is that it conserves the energy of the system. So because this is a linear case, so we can derive an analytical solution for this problem. And to validate the results, we did a comparison of numerical and exact solution, and we found that this is in a good agreement. So it means we are on a good track. We can make our model more complex by moving from linear to nonlinear shallow water equations. So the problem now looks like this. Another thing that I want to mention is that in shallow water equation, we are solving fluid problem only at the surface of the fluid and not for the whole domain. So that's why you can see in the picture, it's just the surface of the fluid and not whole domain. And because now it's nonlinear, so it means now the movements are big enough. And if you pay attention, then you can find, let's say at time 0, the wave maker is at 0, and the length of domain is from 0 to Lx. But after some time, when wave maker is at LW, so it means the length of the domain is from LW to Lx. So it means it's shorter now. How do we deal with a problem like this? So it means either we should have a time-dependent mesh, like we generate a mesh at each and every time step because the length of the domain or dimension of the domains are changing. So this is a problem that we faced. And then because if we generate mesh in each and every time step, then it will make the model expensive. So it's not solving the problem. So what could be the solution? The solution is to transform or map the moving domain to a stationary domain. Solve your problems for that stationary domain and then map it back to the moving domain. So all the computation is done in this stationary domain. And then when we show the result, we map it back to the moving domain. So you can see actually waves being generated. And these are the transformation equations. And the results that we got for these problems are here. Actually, if you notice, we have generated an interesting case. At first, you see that time 0, you see a solid line, which is green. It means that initially free surface is at still, no waves are being generated. Then we turn on the wave maker, let's say for one time period. So this dashed blue line, which is the wave which is being generated. And then we stop the wave maker after. So you see that this blue line, it actually moves forward with this red dotted line, which is like final time step. And then because no wave is being generated, so it becomes flat. So you can actually control the motion of wave maker. You can turn it on, turn it off as you want to generate your waves. Now we can solve our problem for moving domain and all at the surface of the fluid. The next step of complexity is to solve it for whole domain. So now we need a different model, which is potential flow model. And the specialty is that it can solve for whole inner domain. It considers every part of the domain. And if you pay attention, you can also see that the motion of wave maker is also complex. Now it's angular, it depends on y and t. And the good thing is now this model is very much close to the experimental facilities. And for this kind of problem, we have experimental data to do the validation. So we got some experimental data from Maritime Research Institute, Netherlands, because they have these facilities with different wave makers. So they generated some experimental data for us, and we run the simulation for the similar conditions. This is a piston wave maker. At T0, you can see free surface is flat. No waves are being generated. When time is T1, you see some waves. So these are just the evolution of waves through time. This is the result from different probes at different length of the wave tank. And we found that the experimental data and numerical data, it agree well, it's in the acceptable range. So for different probes, you can see at 10, 20, 40, and this 50 and 54, the agreement is quite acceptable. Similarly, we did a comparison with another researcher, Gagarina, and we found that our model generates results which are in the acceptable range of the results that Gagarina got, and also the experimental data. So now, the final part is to implement the wave-flap wavemaker. And this is the problem on which I am still working at the moment. So I will just give you an overview how a wave-flap wavemaker looks like. The specialty of this kind of wavemaker is that it can solve fluid problem for deep wave. So for the potential flow model with piston wavemaker, it's good for the wave tanks, which are not very deep and are longer compared to the depth. But for wave-flap, it can also solve the problem for very deep. And it's like to mimic how the structure, the waves will be behaving for deep ocean. So this is my new challenge, and I'm working on it at the moment. Thank you.
Thank you very much, Marijiha. Okay, and our last speaker today is Maria Takari. She's going to talk about learning the groundwater operator.
Slides. And if you just stay here, this is where the mic is.
Okay, thank you. Okay, good afternoon, everybody. I'm Maria Luisa, and I'm presenting a learning the groundwater operator. So I'm a PhD student, I'm a second year. My PhD is on physics informed deep learning for groundwater prediction. So my supervisor are from School of Civil Engineering and School of Computing, so Chen from Civil Engineering, and then Hevang, Professor Jimak from School of Computing, and I have an external collaboration with the Dutch Research Institute. In this presentation, I will show DeepONet, so this particular neural network as a surrogate model. I will give some good and some not so good results, and then finally I'll resolve the not so good results and some future work. So the challenge, the main kind of the problem that's that initiated my research is the fact that the current groundwater model is can be quite slow to run, especially on a large temporal and spatial scale. So it can actually take weeks for a model to produce results, which if we think about climate change and wanted to have maybe a long-term scenario and for a national scale, that's this computational expensive, the fact that the model is computational expensive can be a bit of a bottleneck. So you can see an example on the left of the chalk aquifer of Yorkshire. So you have different points in which have abstraction of water, and then you can imagine you have different geology, and the geology actually governs how the water moves. So eventually water tends to go where it's more permeable, where it's easier to flow. On the right-hand side is how I translate it for an abstract for scenario. So eventually I have on the left location of a pump, which in this case I place in the middle, but can be everywhere. And then on the middle figure, there is a hydraulic conductivity, which you can think about like as different type of materials. So eventually how permeable the material is and where eventually the water will flow. And on the right-hand side, you'll see a snapshot of a hydraulic head. So eventually where the water is and where it's flowing in time. So can I build an AI, fast AI? Can I use AI as a surrogate model? So eventually, a couple of years ago, in 2019 and 2020, two models were developed. One is called the Fourier Neural Operator, and the one is called DeepONet. And they received quite a lot of attention in the press as being the neural nets that can solve the world's hardest mathematical equation, were blazingly fast. So I decided actually to try with that, and I decided to, among the two, I chose DeepONet because it's actually based on a mathematical theorem. So it also gives that the nice theoretical foundation. So how does it work? DeepONet learns an operator. So if you're familiar with neural network usually learn functions, but an operator is a higher level. So eventually it learns the operator G, which receives some input function U. So eventually what you see here is on the right-hand side is what the, so is G that depends on the input function U. So that's the prediction of the network in some point, for example, spatial point X and Y in location P. So there are two neural networks. One is called the branch and the other one is called the trunk, and they multiply together and then they give you the results, which is G. The input to the branch is the input function, and the input to the trunk are the location. So in this case, a spatial, but can be spatial and temporal location. So how do I translate this mathematical setup in my case? Well, in my case, let's start from the input function. They are the pump location and the hydraulic conductivity. And then the location are just points in my domain. And my output function is the groundwater. So in this case, is in one time step, but eventually that's going to be the evolution of time of the groundwater field. But eventually, DeepONet doesn't need to see the whole solution, but it's because the G depends on X and Y on location P. So it's actually only see the solution and learns from the solution only a sparse location, which is actually really nice because in real cases, you only know the solution and you have observation only on sparse locations. In my case, I'm training with synthetic data, but still, a next step could be to actually apply in a real case scenario where we only have these sparse observations. So in some first analysis, I only so the only things I would change, I would put the pump in the center of my domain, and I would only change the hydraulic conductivity. So the input to the branch net is only on the hydraulic conductivity field. And I created some random field for hydraulic conductivity. So you can see some example here where I can have different. So I create a random field and then I discretize different classes because the soil or materials are actually have very strict boundaries. So for example, I can have 7, 15, 1 classes. And so I train the DeepONet and I actually get really good results. So you can see an example on the right-hand side. So you have the first image is the target, and then I have the middle one is the prediction of my network, and then the difference between the two. And then on the bottom, I have some results that show, for example, going from the first one is for the first group is about 1 time step, and then I go time dependent or 32 time steps. And then I show the difference in actually the amount of points that I sample, because indeed, as I told you, I'm not looking at the whole solution, I'm sampling points. And of course, when I go time dependent, I actually need more points because I am an extra dimension. And then the different colors are about the different type of distribution. So if I test on in distribution data or out of the distribution data. But eventually, summing up, results with when I only change the hydraulic conductivity are actually really, really good, and the network can learn very well. And then I did a next step. So I decided to also change the location of the pump and put it everywhere in the domain. And what I would see is that the network wasn't able to learn, and the results were actually really, weren't good. So I did a simple test, and I trained the network with the wells. Instead of putting the well everywhere in the domain, I would put it only on the top or so at the north and south position. And then I would test with sample where the well is in the middle. So what you can see is that in the training data, so you have still the target and on the left and the output of the network on the right. So the network would actually learn well on the training data. So it would actually memorize the training data. But when I go on the testing set out of the distribution and I put the well in the middle, then the prediction of my network is actually an averaging behavior of what is seen during the training. So that means that the network isn't learning, but it's just memorizing, which of course is not what we want for generalization. So eventually a lot of trying and errors, and I got to this current setup, which is the one that works. So instead of giving, for example, at the beginning, I was giving a binary image where 1 is the location of the well and 0 everywhere else. I need to give a smoother image. So this kind of a type, I call it proximity to the well, something like a distance map. And then I also need to add the coordinates of the well as an input. And then I have to give that same information of the distance from the location to the well also for the when I actually have my query locations for the output. So I actually need to to change the way I give the input and also to condition the network. So to actually give kind of an extra information and give quite a few times the same type of information in different in a different format. And then, yeah, and now I finally get good results. I'm still not fully satisfied. They are still not as accurate as when I only change the right connectivity. So it's still a work in progress. But yeah, the main conclusion is that I have to find the right input representation, which is the key for good learning. And also that this DeepONet needs to be adaptive. And something I'm also looking at is maybe changing the architecture. So not only change the input, but also adapting the architecture to my specific problem. And an open question is the fact that eventually so if I go to the previous here, I'm giving the location of the well also to my trunk net, but according to the initial conventional DeepONet, the space of the function, the input function is only on the branch net. And I would like actually to stick to that, to stick to the conventional DeepONet. An open question is what are actually maybe the most effective. So maybe I found an effective representation, but is this the most effective? Or maybe I need to change the network architecture so that I can actually be still close to that original mathematical formulation. Yeah, in summary, I show a DeepONet, how we can use DeepONet as a surrogate groundwater model, some good and not so good results, especially some good results when I change only the hydraulic conductivity, and not so good results when also the location of the pump is changing. And then I actually resolve it with this type of inputs and some open question for my future work. So thanks for your attention.
Thank you very much. Thank you, Maria. I'm going to put up Megan back, so we have some time for questions, which is great. So I'm just going to quickly show. All right. Megan, are you still with us? Hello. Okay, so we can take about 3 questions for all our speakers. Maybe the speakers who are here, maybe you can come here, so then people can ask you any questions. Are there any questions from the audience to any of the speakers? Okay, Meg, maybe then I'm going to start. Megan, there's a question for you. I was interested to what extent the Bradford City Council is actually maybe using this really nice tool that you have developed, and maybe how it is using it. Thank you.
So currently, Bradford City Council are providing a lot of the data, so particularly for education. So education that's not necessarily held by the Department of Education, but more local level from each school. And because this is quite early stages that we're just building the data sets and building the databases, they haven't actually yet been able to use it. We've not given any data out to anyone as of yet, because we are still fine tweaking them before we let other people have access to them. But I think hopefully we're looking at working together with Bradford City Council to look at things like I spoke about earlier, so things like autism, they're very keen on some reducing waiting lists and things like that. So hopefully they'll have access, they will be able to work collaboratively with them. But they've been a key partner so far in actually letting us have some data from them.
Brilliant. Thank you very much, Megan. Any questions from the audience? No? Maybe then a question for you, Maria, and Payman, because so I noticed that you're both using a version of the neural networks for kind of doing your analysis. I was just wondering why you specifically chosen this method, and whether another potential approach could have been used for doing your analysis as well. So maybe we can start with you, Maria, and then I'm going to give it to you, Payman. Yeah.
Yes, eventually last year I used a different method, which is UNet, convolutional encoder-decoder method, and which is quite nice when you have spatial images. But I think, for example, one reason for choosing, well, I think I'm still in exploration phase, just because all these neural networks are so, and this physics informed neural network is such a young field. So that's why I kind of, so I developed that method, and now I'm actually looking at this other method. And then another reason why I chose this method is, because it's based on this mathematical theorem, and also because of the sparse data. And I think, that's, but there are several methods. I think it's very active field of research. So it's, this might not be, and probably won't be the best method, but yeah.
That's a good question. Actually, that's a philosophical modeling question. So we have the principle of parsimony in modeling, which says that you have to always start with the simplest form of the model to, because to enhance its prediction ability. So the reason that I'm using neural network is just first is for the computational expenses of the process-based model, which is a numerical model. And it can even run when you run it randomly. It can run to areas of the domain of the input parameters that is not solving, is not giving you any result. So that's one problem. When you use you don't have that problem once you feed the neural network to that model. You don't have that problem with the neural network. It's always wrong. And so the thing is, they have a thing of people say that garbage in garbage in modeling. That's so it also depends on what kind of data you use. So when your data is actually generated by another model, your data is smooth. So usually the simplest form should work. So I use the simplest form of the neural network. It's just a simple 3-layer perceptron. I don't need to use the deep layer, because it's basically a regression problem also eventually. And when you use actually, when you want to develop this, the process that I have developed is to maximize its prediction ability. And to do that is to, we have the validation set and the test set. So if the test set is good compared to the validation set, then that's the prediction ability of your model is high. If you usually, try the model for different number of nodes, because the number of nodes, the hyperparameters of the model are known initially. So you have to, run the model and, test it for different cases to get the, the best case for that number of and the optimum is where the least is, and the best, ratio of the test set to the validation set. That's the maximum prediction ability of the model, I think.
Thank you very much. Last chat. Oh, yay.
I'm not really, I've heard of deep ONet and operator type networks, but I've not looked at them at all. It sounds really interesting in terms of, it sounds like it'll be more related to the physics in the end. and you'll get something more like that out. But how do you know what it's done in the end? Is it explainable? Do you know what the operator looks like? How much information does it give you?
Thank you.
Thank you. Well, it's still a neural network, so you have it's still a black box at the end. But there is a similarity with the finite element method in the sense that the trunk network are the basis function, and the branch net are the coefficients. So eventually, the DeepONet can be seen as a general form of so a finite element can be seen as a particular form of what DeepONet does. So eventually, indeed, DeepONet is used to solve, and what I'm doing, I'm solving partial differential equations. So it kind of is a way to solve the equation. Another way that a step further, if you want to get more insight and you want to have, which is also something I'm looking at to get to enforce the physics. There are different ways. I mean, I'm using a regression loss. I'm just purely learning from the data, but you can also add a physical term of the loss function. So, for example, it's physics informed neural network. Or maybe try to understand a bit more, eventually look more into the architecture or change a bit the architecture to kind of enforce the physics. But yeah, there is still a neural network, so you won't get.
But it's inventing the rules of the system.
Yeah, kind of. Yeah. So that's it's a general, and also the Fourier neural operator, the other method that was developed. So this was developed by Brown University and Fourier neural operator by Caltech University. Actually, DeepONet research group showed that Fourier neural operator is also a subset of DeepONet. So it looks like a very general framework.
Okay, thank you very much. Maybe just before we finish, because we just have about 1 minute, is there a question maybe for Vajiha as well? Otherwise, I would have one. I think it's really fascinating. I was just wondering whether, like, this kind of simulations models could be used, for instance, to a kind of advanced research on a tidal wave energy generation. Like, like there are attempts at the moment to try to use tidal waves to generate renewable energy. So could your models help with that?
In fact, my real project is fluid structure interaction problem. And for that, I'm first developing this model to generate waves. And the next step after WaveFlap WaveMaker is to couple the system with a structure. So if you add a buoy or any tidal energy extractor machine or whatever, then it should be coupled with the water waves. So it's a fluid structure interaction problem. And that's what my final goal is. After WaveFlap, I will be including a structure. So you can just change the shape of this structure and some boundary condition, then you can in fact model something like that. Yeah, in fact, one of my senior, he's working on using similar model to model tidal energy system. So yes, it can be extended to that. Thank you.
Okay. Thank you very much. Sorry for dominating the session with my questions. Thank you very much. I think this is the end for today. There will be something social, I think. Is there something social? I'm just going to give over to Alex.
Thank you, everyone. This is the final session for today. We don't actually have anything social penciled in, but feel free to sort of mingle and move on to other places. We're back tomorrow starting at 11, but there will be free coffee from 10. So if you want to come back for session 2 and have a chat with people, come along at 10 and we'll have coffee up in 9.57. But thanks very much for a great first day.